let me introduce myself. My name is Armand, Leroi. And I'm here by virtue of being one of the people who has been here longer than almost anyone. That's not quite true, but so the years go by. And for that reason, I was asked to help organize this event, and I'm going to be hosting it today. It's 75 years since Silwood was bought by Imperial College, and a field site, a field station was founded here. At the time, nobody would have thought just what a powerful and important and influential place it would become in the future years. But so it has become. And so on the 75th anniversary, we decided that we wanted to reflect on our heroic past, all the things that we've done in the past. Think about what we're doing here now, in the present time, and also consider what we might want to do in the next 75 years of ecology and evolutionary biology. And to that end, we've been doing a variety of things. We've got new exhibitions, and we'll be showing a film, and so on and so forth. But really, it's about today, where we have an opportunity to discuss those things. But the real thing that we wanted to do was to see so many of you again. The alumni of Silwood Park have spread throughout British ecology in departments right across the country and across the world, and we've had literally scores of visitors who have come here, all of whom have made Silwood Park so influential. And we wanted to see you, our old friends again. And for that reason, that's what this is really about. And I want to say, welcome. Silwood's distinctiveness is from its mother department in South Kensington, is not just geographic, it's also intellectual. And for that reason, we have often felt the need for slightly separate governance over here. And we have, in fact, a deputy head of department who's head of Silwood Park. And that is Guy Woodward for the last few years, and he's going to say a few words.
Thanks very much, Armand. Can you hear me okay through the lectern microphone? Okay, great. Well, thanks very much. It's an absolute delight and an honour to see all of you. So welcome, one and all. I would also like to say that in the 75th year, we've had a few preceding events sort of building up to this. These have included a panel debate. We also have a lecture, which will be part of a sort of roadshow which goes into South Kensington. But this event tonight is particularly a focus for our community, our connections, as Armand has said, close to home, but also further afield on the global scale. So thanks very much, everyone, for coming. I hope you enjoy the fantastic programme. You're more than welcome, and we look forward to chatting with you both formally during the meeting and informally with drinks and so forth at the end. Thank you. I'll hand over back to Armand.
Thanks, Guy. When I first met him, when I interviewed here in 1996, it was a beautiful spring day. We were sitting outside with Mike Hassel and Charles Godfrey, drinking a pint, as you do by the refectory. This chap came over, introduced himself. He said, "Hello, Mick." I thought he was the gardener. I mean, it was a natural mistake to make. I mean, the inference was obvious. I mean, the man had muddy boots and muddy clothes, and there was dirt under his fingernails. And he had this sort of soft, sort of regional burr, very different from the Oxonian tones of Mike and Charles. And I thought, here I have, I had just come from New York, by the way. I thought, here I have an authentic English, man of the soil, sort of a Thomas Hardy type. He's just been out there doing his agricultural thing. Anyway, the gardener then began to ask me about my research. And I thought, crikey. Even the gardeners at Silwood, are throwing Latin binomials around. This is a pretty amazing place. Anyway, Mike quickly corrected me that I was, in fact, talking to a very distinguished professor of plant ecology. I don't have to tell you much about Mick's accomplishments, but all of you will know it. Let me just try to break them down into three. There is first, of course, his scientific work. I'll just mention two aspects. The first is his work on plant-animal interactions. Before Mick, there was an ecology of plants, there was an ecology of animals, but Mick brought plants and animals together and built a new field. Later, in the 2000s, he did the same thing for biological invasions. He really established these fields, and both of these have major books by which he did so. This is quite independent of the dozens, hundreds of papers, which he's authored. Even if you're not an ecologist, however, it is probably the case that Mick has directly impacted upon your life and upon your work. And he has done so via his teaching of statistical computing. He wrote, of course, a book in the 90s, which was GLIM, and then there is the R book. The R book, which we have all learnt to love and loathe. This is my copy. You can see it has been literally read to bits. It does have abominable binding, but that's not the point. Mick taught us how to do statistics. He'll be modest about this. He'll say he didn't invent the languages, he didn't invent the tests. But without that book, most of us would not be able to have done the research that we have done. The influence has just been immeasurable across the world. There's one more thing that you will sort of know about him, but that is quite independent of this. And it's worth making the point, is that he is probably Britain's, no, he is not probably, he is Britain's preeminent botanist. He is the author of the floras of Berkshire, East
Sutherland.
Thank you. East Sutherland, St Kilda, and the author of the forthcoming Flora of London. There's quote marks around that. There's some issue about its name. London is divided into 3,500 one-kilometer grids, square grids. Mick says he has walked all of those 3,500 square kilometers multiple times. He has contributed more than 1 million records to the British plant databases, more by far than anyone. This is a nation of botanists. If he is our greatest botanist, then he is very likely one of the world's, if not the world's, greatest botanists. But he's more than that to us. Mick is the heart and the soul of Silwood Park. If I mistook him for being the gardener of Silwood, it is a thing that he would have happily acknowledged, because in fact he is. If you look out of this window and you see the grounds here, the planting, the landscape, that is largely Mick's work. If you see the great experimental plots, that is Mick's work too. And he's also the memory of Silwood Park. And it is for that reason that we've asked him to talk to you today. Mick.
So this is a personal history of the place, and it's a partial history of the people here. So if I don't mention your favourite Silwoodian, please forgive me. I apologise in advance for any omissions. The hero of the piece, without doubt, is James Monroe. We would certainly not be here together today were it not for him. So here he is. He was the professor of zoology and applied entomology from 1930 to 1953. Uh-oh. Interesting. So what was it about James Monroe that was so important to us and to Silwood? Well, he was a superb administrator to begin with, and he had friends in high places. This turns out to be important. And he was a member of this place here, on the corner of Piccadilly and Waterloo Place, on the site of the original Carlton House. This is the Athenaeum, which was the intellectuals' club. You know, you had political clubs, and you had military clubs. This was the intellectual club, and he was a member of that. So in the 1930s, it was obvious there was going to be a war, and Monroe was advocating that the government spend money on research on stored products, because in the event of a naval blockade, we'd be very short of tobacco, fruits, but principally wheat. And under Monroe, Imperial had become the foremost place for studying the use of pesticides inside buildings. And that research was carried out somewhere else. Imperial's biology field station in those days was in Slough. It was a bit like Silwood. There was a house and plots for experiments. In the background there, you see the A4 road with Slough on the left and Langley on the right. And that was Herworth, bought in 1929. And during the war, when they were carrying out the research, the government took over Herworth. They basically kicked Imperial College out of their own field station. It was taken over by the Department of Science and Industrial Research and became the Pest Infestation Laboratory, Slough. So at the end of the war, Monroe was looking for a new home for his Imperial College entomologists, and he had money to spend because the government did compensate Imperial for booting them out of Herworth. That's what Herworth looks like today, and we just hope that Silwood doesn't befall the same fate anytime soon. So what did he do? Well, crucially, in 1947, he found Silwood and managed to buy it, and that was the first 80 acres. Then he had to get rid of the army who were in here. It was a military hospital, and they were reluctant to leave. So that's where his friends in high places came. He bumped into people in the Athenaeum who knew the leader of the army, who told the commanding officer here to go. So that was very important. Then a year later, he helped to buy Ashurst on the other side of the property, another 22 acres. And the idea here was that Ashurst would study insects and pesticides, and the living insects would be studied on this side of the campus. And then in 1953, Monroe bought Silwood Farm, which is a much bigger addition to the acreage, another 127 plots, plus Silwood Lake, which was 6 acres. Now, why did he choose Silwood rather than any of the other military-occupied big houses and estates that were available at the end of the war? The answer was this: the 701 Green Line bus. Every hour, it stopped outside Silwood, and then amazingly stopped right outside Imperial College on the steps of the Royal Albert Hall. So it was fantastically convenient. And apparently, that's what tipped the balance. So here we see post-war Silwood as Monroe bought it. So let's go through this in some detail. Here's the manor house, more or less unchanged. Here's South Gravel, that became the Science Park. Here's North Gravel, absolutely covered in nissen huts, that became the residences. Here's the refectory, that was the sergeant's mess. This is where William Penny Hall was eventually built, and this is where the nuclear reactor was eventually built. So this is very grand, isn't it? This was the original house on the footprint of the building you can see through that window there. It was built during the enclosures, 1788, by Sir James Sibbald. I'll mention a little bit about what kind of guy he was and what he did later on. But 90 years later, the guy who bought Silwood didn't like that. He didn't think the rooms were big enough for the kind of parties that he wanted to hold. So Charles Stewart built Silwood specifically to have parties, particularly also during race meetings, with all sorts of shenanigans, which I'll go into a little bit later. That might look vaguely familiar. The architect was Waterhouse, who did famously the Science Museum in South Kensington. At the time, in the late 19th century, he was Berkshire's foremost architect. So that building basically was the entire Silwood life at the time. So because it was built for parties, it had extremely extensive cellars. And those cellars in the basement were the home of the insects in their controlled environment rooms. On the first floor, there were all the big function rooms. So this window here is the lecture theatre, the library's at the back. This is a very important building in Silwood history. This is where we had coffee in the mornings and tea in the afternoons. And they were a big deal in those days. Everybody went. Staff, students, ancillary staff, and so on. Big deal. The science basically went on this floor. So this is my castle's office window here with a lab next door. That's the window out of which I looked when I did my PhD. So this was basically the university bit. And in the roof, in what had been the servants' quarters, was where the students lived, often 4 or 5 to a room in those days. Imagine that now on your prospectus. you will enjoy sharing with the following people. So that's what Ashurst looked like when it was bought. This is where the insecticide work was done. And from the air, the estate at Ashurst looked like this. There's the house. There's the stable block. Here's St. Michael's Church and the graveyard. You see the graveyard was smaller then. There were fewer dead people in those days. This is interesting. This line of veteran oak trees here was the line of the old road that went from Ascot to Beggar's Bush, which is the Cannon Crossroads. And Sibbald didn't like this road going right across the back of his brand new house. So what did he do? He moved the road. That's what you did in 1788 when you were rich. So there's the old line of the old road, and here's the new one. The London Road is here, the Morris Convent is there. So he just moved the road. Silwood Farm was bought in 1953 and brought with it the biggest extra chunk of land. Here's the farm in the foreground. The manor house up here, the ha-ha, the oak tree, which is the memorial to the storm of '87, and the lake, designed and built by Humphrey Rempton, the famous landscape architect. The story goes that Sibbald originally intended to build the house here, next to the original manor house that was called Eastmoor. But when he came here with Repton to sort out the site, the smell from the farm and the associated tannery pits was so disgusting, he said, I'm not going to build a house here, despite the fact that by that time Repton had spent all this effort building this lovely lake to look at from the house, on which were going to be moored quarter-scale painted Chinese boats to make the river look even bigger. Anyway, so that never happened, so that happened instead. So when we bought the farm, we got Nash's Copse here, the bit of ancient woodland, water meadow. The boundary ran like that. So this was part of the original Silwood purchase, and you can see the Nash's Field plots there. So let's think about Silwood as an outdoor laboratory. What have we got here? Why was it so important for biodiversity research? Well, the first thing was the lake, but there are also these extremely interesting red streams feeding the lake, and they're red because of the iron bacteria that live in them. All sorts of interesting research on the mobility of iron bacteria was done here by Michael Carlisle. He discovered that if you tipped a magnet over next to a microscope that you're watching these bacteria down, they flipped as you moved the magnet. Amazing. We've got woodland, which in springtime is beautiful with bluebells. We've got grasslands that are stuffed full of long-term experiments on herbivore exclusion and nutrient addition. We have wetlands. This is a link with Richard Southwood. This plant in the foreground here is Scirpus sylvaticus, and Dick discovered a heteropteran, a bug, new to science in Britain on that plant. And we've got arable. It's an extremely rare community in the Ascot district these days, simply because the soils are so infertile, you can't do profitable agriculture. So you do profitable housing estates instead. So one of the things that affects the landscape immediately around us at Silwood more than anything else is the beautiful arboretum that we've got. In that arboretum, there are 123 champion trees, according to the tree register. And of those 123 Berkshire champions, 10 are British champions. So the biggest trees of those species in the whole of the British Isles are right here in Silwood Park, including this fantastic thing here, which is a birch called Betula dahurica. The other thing about the management of the estate at Silwood is we were way ahead of our time. We were rewilding here 40 years before it became fashionable. So what do I mean by that? I mean, we stopped using pesticides, we stopped using herbicides, we stopped using fertilizers, and we started allowing nature to take its course when we had disturbed ground. Instead of landscaping it, we let nature take its course, and we allowed the succession to produce whatever ecosystem it felt like. So I think that's the essence of rewilding, is you don't have a target and manage to get there. You do something and leave it completely alone in the meantime. So let me tell you about how the research focus changed in the two places, starting with Ashurst. So the original people, Page and Lubati, were proper insecticide chemists. They were working in the era of DDT, and they were looking for new insecticides, pyrethroids for knockdown, and much more subtle systemic insecticides to make the plants toxic to their pests. Then in 1963, with outside funding, the Shell building was erected, and that started a brand new era of parasitology. And the parasitology fell into two broad camps. There were the protozoologists and the nematologists. Top right there is Cyril Garnham, the greatest living parasitologist, much better than being the greatest dead parasitologist, I think. And nematology had Professor Bernard Peters, who was a pioneer in agricultural nematology. So that's what Ashurst was in the intervening years. And then through a real coup of recruitment, Dick Southwood managed to recruit these two extremely eminent entomologists from Cambridge when Vincent Wigglesworth's Institute, the Agricultural Research Council Insect Physiology Group in Cambridge, closed in '67. And Dick Southwood managed to convince these two, who I'll just mention in a minute, to come to Ashurst. So on the right here is John Kennedy. He was famous as an extremely critical thinker in insect behavior. And what he studied was the behavior in relation to flight, and particularly into movement and orientation. Tony Lees, on the left, was interested in insect clocks, in photoperiodism, and in hormones in insect development, particularly the determination of sexual morphs in aphids. Two extremely influential entomologists. What about Silwood? Well, in 1947, when we started doing things, the main figure in entomology in Silwood was O.W. Richards. That was his name, but everyone called him O.W. uniformly. Now, it's interesting just to think a bit about his background. He went to university in Oxford, and these were his teachers. Now, just think about that. He's being taught by E.S. Goodrich, Julian Huxley was his tutor, Sir Gawain de Beer taught him, and J.R. Baker. These were seriously good biologists. And then those were his teachers. These were his colleagues, his peers. Charles Elton, one of the most famous animal ecologists of his generation. Neville Wilmer, E.B. Ford, the butterfly man. Ruth Deansley. She became Lady Parkes because she married Alan Parkes, who, amongst many other distinctions, discovered that you could freeze semen. So he revolutionized the animal breeding industry, amongst many other distinctions. All of those people who were later to become extremely influential thought that O.W. was without doubt the outstanding entomologist in his generation. The other person who came from Slough in '47 was this wonderful lady. This is Nadia Valov, a chain-smoking Russian emigre. Fantastic lady. And her expertise was in Ochinarynca, these things on the left, froghoppers. Now, between those two people, they really revolutionized the study of insect ecology in the field. They published keystone papers from the fauna of Silwood Park on grasshoppers, on ants, on the broom beetle, but most influentially, what they did for the first time was to study plant-insect population dynamics in the field using insecticides to exclude the insect herbivores. And that experiment was big. This is what it looked like. There's a person for scale. In the background, can you see in the distance, you can see the houses of Cheapside, because Nash's copse hadn't recovered from having been felled before it was sold to us. So this was a big experiment. And you can imagine, sprayed with insecticide, those broom bushes were producing a lot of seeds. They're very long-lived, and they formed a seed bank. So nowadays, the legacy of that experiment looks like this. If you put a fence up, you get a broom thicket, instantly, essentially, and the rabbits make sure there's no broom recruitment at all outside that fence. Another thing that happened at Silwood that was important was the arrival in 1963 of the Cyrex unit. That building was paid for by the Australian taxpayer, and when Cyrex left in '71, that became plant ecology at Silwood. Up to that point, they'd still been in the Bight quadrangle in South Kensington. This is one of the great success stories of insect pest biocontrol. So this guy here, Cyrex noctilio, had destroyed more than a third of the conifer plantations in New Zealand. So when it turned up in Australia, they were very, very keen to find a way of controlling it economically. And this guy, Robin Bedding, a Silwood graduate, an entomologist, found the ideal biocontrol agent in a nematode, which at first they thought was two different species. One nematode that fed on the fungus that the wood wasp injected into the wood, and another nematode that parasitized and castrated the female wasps. Turned out these were two generations of the same nematode, and it was in a brand new family, subsequently named in Robin Bedding's honor. So this was a fantastic success. Now, this is another era. This is the Dick Southwood era of Silwood Park. On the left, you see Dick doing his boss of the Silwood Mafia pose. He does that very well, doesn't he? That's extremely convincing. On the right, he went on to great things in administration. He became the Vice-Chancellor of Oxford University, the first entomologist ever to hold that distinction. And if you look very carefully, you'll see he's clutching a first edition of Ecological Methods in his hand here. So what did he do? Well, he was here from 67 to 79 in a directorial capacity, and he really saw a change of emphasis at Silwood from entomology to ecology and evolution. But much more than that, there was a decisive move towards strengthening the role of theory relative to experiment and observation, which is where we'd been before. And probably Dick's greatest achievement was enticing Bob May to spend his summers in Silwood Park. So here is Bob. He started out as a very good physicist in Australia and became the world's foremost theoretical ecologist. What did he do for Silwood? Well, his collaborations essentially revolutionized multiple different disciplines within ecology. So with Roy Anderson on disease-host dynamics, with Mike Hassel on host-parasitoid interactions. These were heady years for theoretical ecology in Silwood. Now, this is interesting. Dick was elected to the Royal Society in 1977, and he made a very conscious decision to lead a concerted campaign to increase the representation of whole organism biologists in the Royal Society. So successful was he at that after 1977, the joke doing the rounds was that FRS stood for Friend of Richard Southwood. You can see how successful he was in Silwood on the following slide. This is what he did just in Silwood. Ba-boom. Now, these people have come to be called subsequently the Silwood Circle. Here are some of them. There's John Beddington on the left, Bob May, Roy Anderson. In 2007, at Mike Hassel's retirement due, which was probably the last significant social event in the Manor House before it was sold, which is quite sad. But there you are. If you want to read about what these people did, there's a book about it called The Silwood Circle by Hannah Gay. A really significant thing for Silwood was the arrival in 1989 of a NERC-funded Centre for Population Biology, and we are in it. This is, or was, the NERC Centre for Population Biology. We got very generous NERC funding for 21 consecutive years, 12 core positions, and 75 staff and students at peak. It was a really big deal. They were here basically to study biodiversity and ecological function, and one of the components of that was called the Ecotron, 16 chambers, and we are in the Ecotron, or its ghost. This was the Ecotron. There was a row of 8 chambers down that side and 8 more down this side. So here's the Ecotron team from 1990. In the middle there, you see John Lawton, director. At the back with the beard, you see Saeed Naeem. And at the front left there, you see our lovely Ros Jones, who's still here. Where are you, Ros? There you are. 32 years younger there. So the experiments are divided into 2 parts. So there were the indoor ones in here, in the Ecotron, and there were field experiments like this one. In the field, this was the Biodept experiment, where they varied species richness and species identity to ask a suite of questions about ecosystem function. Now, this next bit is very curious. If you think about asking for permission now to operate a nuclear reactor in the Greenbelt, close to Windsor, you can imagine you might get a raised eyebrow. But this was the mid-60s. This was a Labour government. Universities could do essentially whatever they wanted. And we got a nuclear reactor. Here it is. It's quite dinky. And the accommodation next to it is not palatial. This is where it was operated from. Much later, 1971, they got fancy laboratories, lecture theatres, and so on in the reactor block. Now, to us who were at Silwood at the time, this was incredibly important because, here, look, in that basement window was the computer terminal. And it looked like that. It was an IBM punch card machine, and you punched your cards, and then you put them in another machine that read the cards and sent them down the telephone to South Kensington. And if you were incredibly lucky, you would get your paper output back the next day in a van that plied between South Kensington and Silwood every day. So you were allowed one typing mistake per day. So usually that was on the second card in your deck, and you got the paperwork back, changed that error, and you got, the next day you got back, there was an error in the third card. So it took an extremely long time to debug a big computer program. Now, it's got all the destructive power of a 2-bar electric fire, right? It wasn't a big nuclear reactor, but it ended up with exactly the same security fencing after 9/11 as did Aldermaston and Burfield, where they actually make atomic bombs. There was more amusement, too, because when they did that, they installed rising bollards. Now, who can't be amused by the concept of rising bollards? So it was decommissioned in stages. The research basically stopped before 2012, when it was shut down. It was defueled by 2014, and this became it in November 2020. They actually came with the machines and knocked it down. So that's what it looks like now. We call it the reactor desert. In the distance, you see the Silwood gym and the engineering store behind it. But in 2 growing seasons, there's been some really interesting secondary succession going on there. There's a lot of buddleia there and a lot of grey poplar. And there's already 50 species growing there, naturally. They're doomed to be destroyed. Apparently, the college can't afford to put astroturf down, so the students can't have an all-weather pitch. But if somebody old here and rich wants to donate an astroturf working, there you go. So in 1966, stage 1 was built. And everyone who looked at it thought, thank God there was no stage 2. So this was plant pathology. It was built for Brian Wheeler's MSc course in plant pathology. And I think the most important thing about that building is that it was home to Ted Green. Ted Green was Silwood's foremost naturalist, without a doubt. He was a botany technician here before he went to Windsor Great Park. And he would lead fungus forays in there with hordes of adoring staff and students following his every word. He could tell you a bird was about to be seen. Most people can say, that's a blackbird, but Ted would tell you what you were going to see because he was reading their calls. Absolutely fantastic naturalist. The first custom-built student accommodation, which meant students could move out of that multiple occupancy in the roof of the manor house, was this amazing building. There's William Penny Hall without any landscaping at all. And here it is being opened. There's William Penny on the right, looking very pleased with himself. Dick Southwood in the middle, and Sir Henry Fisher, who's chair of the governing body, on the right. William Penny's an interesting cove. He was a mathematician as part of the team that developed the nuclear bomb in America towards the end of the Second World War. And his job was not an enviable one, I suspect. He had to work out the height at which to detonate the bomb to maximize the number of people it would kill. Very, very odd job to have. So the next residents to be made were Southwood Halls. The future of Silwood in the early 80s was very uncertain. So the college, to hedge its bets, built Southwood Halls so they could be sold in the event of the demise of Silwood as semi-detached domestic residences. So they certainly weren't optimized for student living. Now this funny building down here is called Sandy Ride. There's London Road. There's the Maris Convent. We're on the way to Ascot. And that was bought as accommodation for married students, principally married students from overseas. And it was sold only a few years later to pay for the construction of the 2 Brian and Mary Flowers halls. The last of the halls to be built, this is scandalous. The most recent development for students in Silwood Park was 32 years ago. The students used to call this Colditz. Can you see why? Then in 1996, the manor house failed its fire inspection. So we had to get out. It failed it because it didn't have a fire escape from the southern end of the main hall. So the very expensive new internal staircase fitted. It was rewired and re-roofed, and the re-roofing was extraordinary to look at. Here's some slaters up on the roof of the manor house, completely encased in scaffolding. One of the really terrific things about Silwood are the people who come here as visitors to interact with the resident community here. One of the most important to come was David Greathead, who brought the International Institute of Biological Control to Silwood in June 1981, and they lived in a suite of rooms off the balcony in the manor house. You can tell it's David Greathead because of this. You could key David out on the permanent presence of a pipe gripped in his teeth, firmly like that. And this was very successful collaboration. During that period, a thing called the Silwood Project on Weed Biocontrol started in 1983, because they had a fantastic set of data on biocontrol projects that succeeded and, crucially, failed. So this provided the data for the first serious attempts to understand the ecology of invasion and establishment of alien plants, and that became a major growth area for research at Silwood Park. So one of my castle's great friends was involved with this, Clive Moran, who came in summers from South Africa. He was an expert practitioner in weed biocontrol, having successfully controlled some of South Africa's major woody invaders. Cabby liked it so much at Silwood that they bought the land and built a building. This is it. So this was the UK Centre for Cabby Bioscience from 1908 to 2008, and on the ground floor was the Michael Way Library, which had an amazing library of entomology. That was so successful, they extended it. So in 1995, they doubled the size of this part of the building, and that meant the demolition of the Cyrex unit, which had been plant ecology. So plant ecology disappeared as Cabby expanded. And there it is with this new roof. So if you know where we're looking from, these little trees here are now British champions of 2 evergreen oaks. Cabby decided to move to Egham for reasons we never fathomed. But the for sale sign was up for a very long time. And Imperial College were playing a quite canny game. They were putting an effort in until they realized that Cabby were desperate to sell. And eventually, in 2008, the college bought the building and they converted it for MSc teaching into what we now call the Hamilton Building. So on the right there, where there had been the stacks of the Michael Way Library, there are now 4 lecture theatres. And on the left is now the Silwood students' breakout area, the foyer, if you like. So this was a complete revolution in terms of our ability to accommodate numbers of MSc students. And again, to somewhat mild amusement, we had a new canopy installed, at presumably considerable expense, but not obvious of any function. They did put a cover on it, but the cover was called the condom. And it opened for teaching in 2009. This is the cohort of ecology, evolution, and conservation students about to go in to have their very first lecture in the new lecture theatres in the Hamilton Building, as now is. Let's take a change from looking at buildings and now look at a field site as it changes through the years with different uses. So this started out in 1980 as an enclosure to keep rabbits in. Most of our fences at Silwood are to keep rabbits out, but this fence was to keep rabbits in. And these fences here showed the effect of the rabbits that were in there on regrowth in winter wheat. This was a MAF-funded project on rabbits as pests of winter wheat. And if you look very carefully in the background, some of you will recognize Richard Brown. The next thing that happened to that site was it became the National Sea Beet Genotype Collection as part of the Prosamo project. And there's Sue Brown. She collected all these sea beet genotypes from right around the coast of the British Isles, a major effort. Subsequently, it became used for plant population dynamics. Here are the raised beds being built in 1995 from railway sleepers cut in half and 2 railway sleepers making a square. And then the mixture of the soil in here could be carefully controlled And different kinds of communities of annual plants, up to 7 species, were competed against one another. Here it is working in 1997. What I want you to observe is the excellence of the weeding. So this is the experimental area, and this gravel is completely weed-free, thanks to the efforts of the students, including Gabre Asifa. I don't know how many of you remember Gabre, but he was the world's greatest weeder. And he recorded every species he weeded, he recorded the species of it. And this is probably the best database in the world about plant immigration. Extraordinary. His NERC grant ran out in 2007, so nobody weeded in 2008, so it looked like that. And then in 2011, you can see birch saplings appearing, and now it looks like that. So 14 years later, it's a woodland of sallow, birch, and pine, and we use it to discover within species genetic diversity of woody plants. So in 1985, we sold Ashurst to fund the building of garden wood laboratories, and here they are in skeletal form. The one in the middle distance was called East Block, and the one to its right was called North Block. These names weren't very interesting, so now that's called Lees, and that's called Monroe. And this space here shows how you need a special relationship with builders. We moved the architect's plans of North, West, and North Block in order to accommodate an absolutely gorgeous veteran oak tree, which did stand exactly there and spread like that. Now, none of us were here on the first day the contractors came, and on the first day the contractors came, a man with a very large bulldozer knocked the tree over. So, always supervise your construction. It was opened by Princess Anne in October 1988, and there's Nigel Bell talking to her, and Vic Brown, do you remember Vic? Looking on. Now, next door to that was the Science Park, which was begun in '87 on South Gravel, which had been the parade ground of the squaddies who were living in the house. So you have the wartime buildings there surviving, and the new engineering store, which nobody quite knew why was put there, because it was never used for anything remotely important or interesting. It's still here. They moved it, again, at great expense, below the Impetus gym. Now, the idea was that the science park would be screened from the academic buildings by retaining garden wood. But that was without reckoning on the great storm of October the 17th, 1987, when every single one of those beech trees blew over. So this is what the science park site looked like before they started building it, without any landscape at all. Now, there was a genuine collaboration on the science park, and that was this building here, which was MScan originally. It used the reactor for neutron activation analysis to analyze samples for selected heavy metals. So it was used for forensic research and for commercial research on soil. So that was a real collaboration. On the other hand, the Technology Transfer Center, this thing, was supposed to be a Silicon Valley-like synergy between ecology, Imperial College scientists, and entrepreneurs, so that potentially commercial ideas could be viability tested. It never worked. I can't think of a single example where that actually happened in the Technology Transfer Center. The big buildings on the science park looked like this, and this one has an interesting story about it. This was the biggest of the units, and the lease was taken out by BP Nutrition, who were a major firm, obviously BP dominated, and they were doing GM crop research, developing new crops using genetic engineering to introduce traits like herbicide resistance or pest resistance. But timing. This was exactly the time when Friends of the Earth decided to demonize the entire GM technology. So you saw people outside your local Sainsbury's with signs like this: "Say no to Frankenstein foods," says Friends of the Earth. Now, BP were good businessmen, and they saw the writing on the wall, so they walked away from GM, and they walked away from Silwood before they even moved into the building. So they took out the lease, built this really fancy 4-compartment greenhouse, which we're now the beneficiaries of, but they never moved in, and they never did a single stroke of GM work here. So there's a moral there, I think, for all of us. Other research facilities, some of which went well, some didn't. The lysimeters at Ashurst were very successful until they started using radio labeling for the movement of minerals through the soil blocks. When this was decommissioned, it was discovered to be extremely hot. So it cost many thousands of pounds to decommission those lysimeters. The Met Tower, which doesn't look quite like this these days, originally had a very powerful radar on top of it. And that radar was used in the technology that we experience every morning when we look at the weather forecast. This was the technology for tracing rainstorms using radar. So when you look at the next weather forecast, think that the research behind that was done at Silwood Park. This was a Royal Society grant to build the tower and the radar in 1963. If looking down from the Met Tower, you see these things. You see the telescope and its base and astrophysics. The professor of meteorology lived in a farmhouse, which was a luxurious bonus for him. Now, I'm going to tell you a little bit about Nigel Bell now. Nigel studied the effects of air pollution on plant growth, very important work. And when he started doing it, he was behind the American canteen next to the Cyrex unit, and he had open-top chambers and closed chambers through which he would pass variously carefully monitored pollutants like sulfur dioxide, ozone, and so on. The reason I'm showing you this is very few of us have ever had bricks and mortar as part of our research grants. Nigel was a completely different animal to the rest of us. Not only did he have a building, he had two. So he had dry field laboratory built for his open-top chambers here, and he also had a wind tunnel built by the nuclear reactor. Extraordinary. Now the impetus gym. Now this was a very sinister experiment. This was civil engineers for you. This is in a bunker. And what they've got, these shiny cylinders contain different chemical compositions of cement. And at the top, they're open to rainfall. And the idea of the experiment is to see what happens when rainfall, acidic as it is, passes through cements of different compositions. You're obviously interested in how long your motorway bridge is going to stand up for. And the exuvia were collected and analyzed for the chemistry. But it was a really spooky place. It had no electric lighting inside it, so it had an atmosphere, definitely. More recently, Guy's mesocosms were established in 2015. And here we're looking at multiple stressors. We're looking at the combination of climate warming and chemical pollutants on the assembly of aquatic ecosystems in replicated mesocosms. And more recently still, we've got the aviary where the Lundy sparrows live, and we've got the controlled environment building behind it, both 2017, where the mosquitoes live in complete luxury. One of the really exciting things about Silwood wasn't just the visiting staff, it was the visiting students. So here's one of the heroes of Silwood Park, Graham Matthews, who was in charge of the Spraying Machinery Centre. And at the time, they were operating a spraying machinery course, which had long reach. So virtually every pest controller in Africa had come to Silwood to study how to apply their pesticides. And this was funded by the British Council, a fantastic example of government outreach through the Foreign Office rather than through science funding. They were taught how to apply chemicals effectively in terms of targeting, but also with low water usage, because a lot of the places they needed to apply their pesticides didn't have access to a fresh water supply. The GLIM course, which ran from 2018 to 2014, was a 3-week stats course in February, and all the Silwood students took it, the master's students and all the first-year PhD students. But the great thing was there were about 50 visitors from overseas and from other British universities who took part in it as well. And it used a very esoteric language, GLIM, up until 1999, hence the name, Generalized Linear Modeling, and then it switched to R in 2000. And until we had the Hamilton building, it was extremely difficult to lecture to large groups in Silwood. We simply didn't have the space. The only place we could sit 120 students down was in the hall of the Manor House. So that's what teaching the GLIM course used to look like. That wasn't the practicals, of course. They were a bit more relaxed now. And finally, in terms of courses, we had an Advances in Ecology course that ran from 1994 to 2006. And that was a 3-week, very generously funded NERC course for students, PhD students from Silwood and other UK universities, plus paying guests who are PhD students from other parts of the world. And in the 3 weeks, they studied single species dynamics, then 2 species dynamics, and then community dynamics. What was really terrific about this course was the visiting megastars of ecology from all over the world who came to do guest teaching on it. So we had Bob May, for example, we had Stuart Pym, we've got Bob Holt, repeatedly, who's here in the audience today. This was a really exciting time for the students at Silwood. So here's the first cohort in 1994. Watch the aging of the staff members here. I'm not going to go through it year by year, but this is the first cohort in 94, and here's the last cohort in 2006. So both Munro and Southwood are extremely keen on social life, on fostering social life within Silwood. The Munro Club is this black Nissen hut outside on the footprint of the building we're sitting in right now. And that lasted a very long time, actually. And inside it looked like this. And you'll notice an extremely expensive, proper grown-up snooker table in the foreground. Now, that was moved subsequently to the student bar. And on it, Bob May taught us all how to play slosh, which is a very bad use of an expensive snooker table. The everyday social hub at Silwood was the bar, obviously. And from 5 till 6 on virtually every weekday, you would see staff interacting with students and with visiting speakers and seminar givers. It really was a fantastic resource. But more big set-piece social functions all happened in the Manor House, in the main hall. So there were things like the carol singing at Christmas time, you'll remember, with the amazing punch that Mike used to concoct with assistants. It was a secret. The recipe was a top secret. There were things like Burns Night. There were all sorts of very formal things, but then there were very informal do's too, like Bonfire Night and so on. The thing about the social life in those days was it was very paternalistic. Both Southwood and Munro were very keen that all staff attended all social do's. And now, the next bit is hard to imagine these days. Their wives had to come too. So all the staff and all their wives had to be at all the do's. And this is what it looked like. It's just hard to imagine that happening now, isn't it? So music's always been a big deal at Silwood, and this was our most successful musical production. These are Warble Fly. They made 3 CDs, and amazingly, they played at Glastonbury in 2004. So they were the bee's knees. They were good. Silwood has its own Glastonbury, of course. Since 1999, we've had Silfest on the lawn outside the Manor House, when we had our own open-air rock music event. But without doubt, the thing that was most social most often at Silwood was croquet. So every lunchtime, winter or summer, from 1 till 2, croquet was essentially compulsory. So how would I summarize Silwood Park? 3 things. Obviously, great science, wonderful people, and loads of fun. Thank you.
It comes to the second part of our day. With Mick, we heard something about Silwood's past. I confess it was it was an important talk, which we, of course, recorded. More important than you may think, because the truth is we'll never hear it again. I'm sorry, Mick. Let us hope we will hear it again, but in the event that we do not, we have got it on tape. We wanted to tell you about what we're doing now here at Silwood Park. But of course, I was faced with the invidious distinction, having to make choices between my colleagues, and I thought, oh, I'm not going to do that, right? Why don't we instead let some of our very youngest researchers tell you what they're doing? After all, they're the people who actually do the work, right? The great thing about Silwood has always been the fact that here we have a number of people at any given time who have very similar interests, but somewhat divergent skills that they can bring to any given problem and collaborate and do wonderful things. This is essentially what happened in that heroic age. We still speak of it, like the Greeks did of, the Homeric heroes, and when Bob May and Roy Anderson and Mike Hassel and Mick, reshaped theoretical ecology. And by the way, we've got these boards at the back there where you can read about it. It is that sort of intellectual promiscuity, as it were, from which great science is made. And that brings me to my first speakers, because Tom Smith, nobody here, I think, has been more promiscuous, at least in the intellectual sense, than Tom Smith. So Tom did his bachelor's at Edinburgh and his master's in PhD at Silwood. At Silwood, he's done his PhD with Tom Bell and Summer Power on microbial ecology and temperature adaptation. But he's also worked with Tim Barraclough. And what he's going to tell us about today is a project that is not with any of those three guys, but rather that he's done with Will Pearce and Mike Tristram. And the reason I wanted him to talk about it is because it's part of Silwood's response to the pandemic. COVID-19 came along, Silwood stepped up. It's not if we don't have the talent here. And that's what Tom is going to be talking about. Tom.
Okay. Oh, okay. Maybe I'll stand over here. We're okay. Great. I'm Tom. Normally I work with Tom Bell, but I'm going to talk today about some work I did a little while ago with Will Pearce over there. So, cast your mind back to the start of 2020. This was Silwood. Everything was great. We were out doing our field work, collecting some samples, bringing them back to the lab to do some interesting experiments on them. And then the whole world stopped. We had this global pandemic, something that we'd never seen before in our lifetimes. And a lot of people, well, certainly here at Silwood, everything stopped. And we didn't know how long everything was going to be closed for. We didn't know when we'd be able to get back into the labs and do our normal research. So a lot of people were wondering, well, okay, are there other things we can do instead? Are there ways maybe we can be helpful in the pandemic? Are there questions which are open about COVID-19 that maybe we have some of the skills and expertise to answer? So one such question that arose fairly early on in the pandemic was, do we think COVID is likely to be a seasonal virus? That is, do we expect there to be peaks and troughs in transmission between winter and summer conditions? And so I was part of a team here at Silwood who started looking into this question. We built a sort of multidisciplinary team of people to look at this question. Over at Silwood, it was led by Will over there. I was the postdoc. Mike, I don't know if he's here, but Mike has a lot of expertise on ecology and evolution of viruses, so that was very useful. But we realized that a lot of this work was going to sort of fall into the department of epidemiology. So we collaborated with some epidemiologists. So Lorenzo and Malaria are epidemiologists from Imperial South Kensington department who we collaborated with. And particularly they were involved in Imperial's COVID-19 response team, which are like the people who were developing the models which our government were literally basing their policy on. So they know what they're talking about, and they were sort of there to ensure that the things we were doing, the models we were doing, the results we were getting made epidemiological sense, you could say. And then throughout this project, we ended up collaborating with loads more epidemiologists, and this became this really great cross-disciplinary big project working with lots of people across different departments of Imperial. So back to the question we were asking, do we expect COVID to be a seasonal virus? Normally, if you want to look at seasonality of a virus, you really want this long-term data, right? You want to see how transmission varies over years, see how that is affected by environmental conditions. We started doing this work at the start of the pandemic. There was no long-term data. There was a few months' worth of data when we started working on this. So that immediately begs the question, how on earth do we start testing seasonality when there is no seasonal data to look at? So we instead decided, as a starting point, to look at spatial variation in transmission. That is, are there differences in the transmission rates between places that are colder or warmer, and can we infer something about that seasonality from that? So in order to do that, we needed, ideally we wanted data from a single country where reporting of COVID was relatively standardized across it, and ideally a big country where there's lots of variation in those transmission rates and variation in climate. So we took, very handily, Etty here, who was another of those epidemiologists from Imperial South Kensington campus, had just published this data set on transmission rates of COVID across the different states of the USA. So we took that as our starting point. We basically, very simply asked, do the transmission rates of these different states of the USA tend to be different based on whether those states are warmer or colder? And we found a really striking pattern. So this is right at the start of the pandemic, right as the disease was in its sort of epidemic phase. There's nothing restricting transmission. There's no policy interventions in place, like mask wearing, no social distancing, no telling people to stay at home. And during this sort of time, this snapshot in time, if you like, transmission, R, is much higher in the states which are colder than in the states which are warmer. So that says, that sort of says something towards that seasonality. And then we looked a little bit later in time. We wanted to know what the impact of these sort of interventions that were coming in were. So a little bit later on in the year, all of the states of the US were in something what they called stay-at-home measures, which was the equivalent to us having a lockdown, going out and mingling with other households. And at this time point, transmission comes all the way down. That's the point of a lockdown. But notably for us, all signal of temperature in the data went away. It doesn't matter whether you're in a cold state or a warm state if you're in a lockdown. So we said, okay, we think temperature is having an effect on the transmission rates of COVID, but only in the absence of these sort of strong non-pharmaceutical interventions. And so maybe we can infer something about seasonality from that, but it's not really good enough. What we really want to be able to see is if these transmission rates vary through time with changes in season, with changes in temperature. So we went back to the original epidemiological model that Etty was using to calculate these transmission rates, and it's too ugly to look at, but we can sort of break it down roughly into something like this, which is that daily transmission rates are being calculated for the different states in the US based on a sort of overall mean, which is modified by the strength of those non-pharmaceutical interventions. So whether people are being allowed to move around or not a lot, or being like sort of told to stay at home, the strength of those interventions is sort of the important factor in this model. And we said, okay, we've got this model. What happens if we add temperature to it? Can we add a new parameter for temperature, fit it to this extra data with temperature, and see if temperature comes out as a useful thing to add to this model? So we did that. We fitted it to the data. These are some of the important parameters in the model. Temperature over here is significantly negative. So in this model of daily changes in COVID transmission rates, when it is cold, transmission is higher. And we wanted to then go a little bit further to validate this model a little bit more. This is basically the sort of data that the model is ultimately being fit to. So this is for New York State. The gray bars are daily deaths in New York State. That gives you sort of a stark reminder of like how bad this pandemic was at the start. That's 1,000 deaths a day in New York State. And so we fit our model across these few months of data up to that sort of dotted line you see. And then for the subsequent 2 weeks into the future, we sort of use the parameters from the model to predict how many deaths we think there'll be, given that we know what the temperature on those days is, and given that we know how much people were moving around on those days. And then the predicted deaths of the model are in blue, and the observed real deaths that occurred over that time period are in red. And it looks like it does a fairly good job. And we did this across all the different states of the US, and we found really good correlation between the predictions of the amount of deaths there will be in this sort of 2-week forward predicting period and the observed deaths that actually occurred. So we've been able to add temperature as an effect in this epidemiological model. It's come out as a significant factor in COVID transmission rates, and we've been able to validate this model and say it's useful for predicting forwards as well. So the sort of take-home messages I want you to get from this are, so we found that COVID transmission rates are correlated with not only spatial variation in temperature, but also daily changes in temperature. But the most important factor in all these models is the non-pharmaceutical interventions. When those interventions are strong, the effect of temperature doesn't matter, so you can always mitigate the sort of high transmission rates of COVID by policy interventions. And then briefly, what are the sort of mechanisms then that can make a disease like COVID be seasonal? Well, there are direct effects of the weather itself on the virus. When it's warm or when there's high levels of UV, virus particles sitting around on surfaces don't last for as long. When it's humid, the sort of droplets you spray out of your mouth, very pleasant, I know, don't travel as far. There are effects of temperature and climate on the host, which is us. When it's cold, you shed more virus particles out of your nose. Again, very pleasant. But possibly most importantly of all in all of this is that it changes our behavior. When it's cold, we tend to have our social gatherings indoors. When it's sunny, we have our social gatherings outdoors. And as we know, most COVID transmission occurs indoors. So although I've shown temperature as the thing we've been looking at in these models, consider temperature as more of like a proxy for all of these sort of effects that happen when we think about differences in season between winter and summer. So what are the outcomes from doing all this work? Well, there were actually loads and loads of studies in the first year of the pandemic looking at seasonality, different data sets, different models, different methodological approaches. But there, all these papers and ours sort of vaguely converged on the same answer that, yeah, we think COVID is probably going to be seasonal, but that non-pharmaceutical interventions are sort of the strongest effect. Please listen to this, policymakers, as we go into the first winter of that first year of the pandemic. We were able to disseminate this work to the public quite nicely. So The Guardian ran an article that was based on our study, and so it was important for us to be able to sort of speak to journalists and make sure that we were getting the right message across and things weren't being misinterpreted. You could imagine maybe maybe a more nefarious newspaper who might want to say something like, good news, everybody, it's going to be summer soon, we can all go outside because you can't catch COVID. We very much wanted our stuff not to sort of get in the way of the actual policy messages that were coming out. We followed this up with other work. We've been looking at how different variants might have spread across the UK with climate. And finally, throughout all this, we've been trying to publish our data sets, curate our data sets, make them open and accessible by other people, and so other people can use this data going forwards. And so I'll finish there. That's basically thanks to everybody who have variously helped across this project. Thank you for listening.
One of the very first people that I met when I came here in 1996, he was actually introduced to me sort of as an inducement to myself to come here, was a lecturer called Austin Burt. He was, like me, had a bit of an American-Canadian background, and we were very much the same age and very similar intellectually, sort of, except there were differences. Austin had a reputation for sort of taciturn brilliance. And, but it's safe to say that for a few years at least, it wasn't clear exactly how this brilliance would manifest itself, I think. But in 2003, he published a paper, which, I'll just get the title right here, Site-Specific Selfish Genes as Tools for the Control of Genetic Engineering in Natural Populations. It was not in Science or Nature. This paper was published in Procroy Sock. I confess that despite the fact that I knew Austin so well, I actually missed the significance of this paper, really. I mean, it was complicated. Essentially, it gave the mathematical conditions under which a selfish genetic element could be introduced into a population and spread through that population and drive it extinct. In other words, wipe out the population, wipe out a pathogen, some species or vector, at essentially trivial cost. And just that, completely specifically. So this paper I'll just give you the short version, became the subject of a grant which was funded by the Gates Foundation, and that is now a large international enterprise, Target Malaria, aimed at eliminating, wiping from the face of the earth, ultimately, the malaria mosquito. But the technology is generic. If it works, and we don't know if it will work, there are trials, and I won't even try to tell you where exactly it stands. It can be applied to anything that we please, really. This is going to embarrass him, but I think it's important to say, if this thing works, Austin Bud is going to be Silwood Park's first Nobel laureate, I think. If he is, however, it will be on the back of a lot of other people's work. And in that case, it is Katie Willis. Now, Katie's going to be, she did her bachelor's degree at Liverpool. Birmingham. Birmingham, and then did a master's degree here in theoretical systems biology in South Kent, and has done a PhD now just with Austin, and it is a computational project. It is laying the computational basis for making sure that this thing that they are inventing doesn't spin out of control. Katie.
Okay, so thank you very much. So I'm going to be talking to you today a little bit about target malaria and how we are trying to help deal with the problem of malaria using genetically modified mosquitoes. And then I'm going to talk to you a little bit about how my PhD work and now postdoc work is contributing to that goal, and is in particular in discovering new genetic technologies for pest control. So first, let's just remind ourselves of the problem of malaria. So malaria is caused by the infection of a parasite, which is spread between individuals through the bite of a female mosquito of certain species. Now, unfortunately, malaria imposes a huge burden on humanity, and it's estimated currently we have over 600 deaths per year, most of which are in Africa and most of which are in children under the age of 5. So not only do we have many deaths, but we also have over 240 million cases per year, which causes significant social and economic burden to many countries, again, particularly those in Africa. Now, for decades, researchers have been looking at new ways to reduce the transmission of malaria, and many of them have been really, really successful. For example, most of you will be very familiar with insecticides and things like bed nets. Now, unfortunately, this progress is at risk. These particular tools are at risk due to the development of resistance in the mosquitoes to these insecticides. And so really we still need to develop new tools to reduce transmission. Now, many of you will also have seen in the news recently the new malaria vaccine. And this is thought to have a huge impact on transmission. However, even with this vaccine and the existing tools, we don't expect that this will be able to achieve elimination. And to achieve elimination, we think that it's still a combination of existing tools and new tools are still required to achieve that goal. So Target Malaria then are looking at genetic technologies to reduce transmission of malaria. And in particular, we're looking at applying selfish genetic elements or gene drive to the control of mosquito populations. So what is gene drive? So gene drive is a genetic phenomenon which occurs in many natural populations, and it causes the biased inheritance of a genetic trait. So normally, with Mendelian inheritance, a genetic trait is transmitted from parent to offspring with 50% chance. Whereas with gene drive, this can be increased up to 99%. And this means that only with the genetic trait being present in a population at only small, low frequency, it can increase in frequency after each generation and eventually has the potential to reach fixation in the population. So at Target Malaria, we're looking at using this gene drive to reduce malaria transmission, And in particular, we're looking to reduce the number of malaria-transmitting mosquitoes. To do this, we're looking at developing a technology which can spread a modification that will allow will affect the mosquito's ability to reproduce. So by reducing the mosquito's ability to reproduce, there will be fewer offspring in the next generation, and so reduce transmission. Now, we expect this to provide an efficient and sustainable tool, partly because of the gene drive, because you only really need to release a few mosquitoes, and then the gene drive process will allow that introduced genetic trait to spread through the population. But also because these mosquitoes, this technology is able to exploit the mosquito's ability to disperse into different areas. And so, again, by releasing just a few mosquitoes in a local area, we expect that these mosquitoes can move, and so the genetic trait not only spreads in the population, but also spatially. Now, at Target Malaria, we're not just interested in gene drive, but also in non-gene drive technologies. And this is because we're interested in developing this technology in a phased approach. And this is because we want to be able to test and learn about our technology safely before releasing something that has the potential to spread. So we're not the only ones interested in this technology. And recently, there's been an explosion of interest, and with it, many different proposed techniques. Okay, so we have split drive systems, we have daisy chain systems, toxin antidote systems, sex ratio distorters. We have Cleave Rescue, split versions of Cleave Rescue. We have split drive killer rescue, and we have sex-linked genome editors. So with all of these kind of new ideas coming out, some obvious questions arise. Okay, so some of these questions are, how did all of these strategies compare to one another? And of all of these strategies, which might be the best for our goal? Now, with these ideas coming out, we've also, and some of people are trying to build them in the lab, there's been the development of lots of molecular building blocks or molecular tools that enable us to build these things. And so another question is, how might we be able to combine these building blocks in new ways to develop new designs that might be useful? And with these new designs, what might the impact be on population size? Would they be expected to spread, or are they contained? How far would they spread? How long would they persist? How efficient are they? And how do these compare to those existing strategies that have already been proposed? Now, there are potentially hundreds of thousands of different designs that you could come up with, and to test them all in the lab by building each one would obviously be far too resource intensive. So during my PhD and now postdoc, I've been working on developing GBSim, which is a flexible genetic biocontrol simulator capable of simulating the genetics and dynamics of populations through time. And this is in response to the release of genetic constructs, of a huge range of genetic constructs, and monitoring, and we can then monitor the size and genetic makeup of our populations. So these are just some examples of the simulations through time. So on the left, you can see a gene drive, and so you can see here the frequency of the construct when released very small frequencies quickly increases in frequency, and with it, the population size shown there in black decreases. We can also model temporary drive systems where, after the initial release, the construct is able to increase, but only temporarily, before it's eventually lost. And finally, we can also model things like non-gene drives, where after the initial release, the frequency of the genetic construct decreases and eventually is lost. And for these two, you can see that the reduction in the number of females in the population is only temporary, and then it returns to what it was before the release. Now, as I said, we're also interested in looking at how we can develop new designs or new strategies based on the molecular tools that are available to us. And this is one of the questions I was interested in asking for my PhD. So in particular, I was interested in whether we could maintain the efficiency that you get from a gene drive, but have finer control over how far it's going to spread geographically. So the technology that we're looking at the moment to target malaria uses site-specific nucleases. And these are proteins which cut specific regions of the genome in order to, specific sequences in the genome in order to drive or increase in frequency. So one of the ways we can localize this then is to utilize existing genetic variation in populations to limit that spread. So we can, in theory, design our gene drive to target sequences in the genome which are enriched in the target population or at high frequency in the target population, but perhaps absent or at lower frequency in the non-target population. And this allows the genetic construct to spread only in the target population, but not reach the non-target population. Now, the problem with this is that this kind of strategy is highly sensitive to resistance. So any of these mosquitoes in this non-target population that carry a sequence that's different to the target, if they move into that target population, they will be highly selected for, and essentially the ability for our strategy to reduce the population size would be far less. So using GBSim then, I explored a range of different designs or of these genetic constructs to try and find that were more resilient to this resistance. And we found, and you can see here, this is the time series again, and we're looking at the relative number of females over time. And we found that if you release 2 interacting constructs rather than just one, we're able to develop strategies that have far greater ability to reduce the population size, even if there's a small percentage of resistance in the population. So here you can see a single construct compared to a range of different double drive constructs shown in the green and the blue. And this advantage really is because we're able to separate out the roles of both inducing some fitness costs and the localization across 2 loci, which essentially reduce the selection pressure for the resistant locus. So GBSim is not only good for just discovering things in theory, but also we can also use it to help design cage trials. So this is an example of some work that I did in collaboration with a molecular biology team in South Kent that are actually developing these mosquitoes in the lab. And you can see here that these are the predictions made by GBSim are in the cream, and then the data that has been collected in the lab in the cage trial is shown in the green and the blue. On the left over there, you can see the number of females in the cages, and that's declining over time. And also on the right, you can see the frequency of our genetic modification increasing through time. So just to summarize, I developed GBSim, which is a genetic biocontrol simulator. And with this, we were able to evaluate and compare a range of different genetic biocontrol strategies, as well as discover new strategies built from many of the existing molecular tools and building blocks. We were also able to design cage trials to test these constructs in the lab. And throughout all of these projects, I think probably one of the most useful or interesting outcomes of GBSim is that it allows us to develop a deeper understanding of this genetic biocontrol technology that we're developing. And not only for strategies as a whole, but also the building blocks that we're using to develop this strategy. And like Armand said, these kind of strategies can be applied to many different species. And the same with GBSim. It's flexible enough and general enough that we can actually simulate many different species, not just in the context of malaria like I've been talking to today. So we can look at other vector species, crop pests, and also invasive species. And so with that, I'd just like to thank my funders, the Bill and Melinda Gates Foundation and the Open Philanthropy Project, to Austin for being a wonderful supervisor, and to all of those people that I've collaborated with from South Kent, and of course Target Malaria for creating such a wonderful environment to work in. And thank you all very much for listening.
when we think about our intellectual history, one of the things that we like to pride upon, one of the points of pride, was the fact that Bill Hamilton was here when he published his great papers on kin selection and inclusive fitness, in which he explained for the first time why it is that creatures sometimes sacrifice themselves for their relatives. It was a breakthrough in evolutionary biology. It reshaped the way we think about evolution. We have a nice poster on it at the back in which we do not mention the fact that he actually did the work at UCL, and that when he did come here, he certainly published it here when he was here. He languished for 17 years without promotion to even senior lecturer. He was an appalling teacher, apparently. So it was only when he only really became famous when he pushed on and went to America. Be that as it may, the thing that I loved about the Hamilton story is that it's about taking an evolutionary paradox, a thing that we do not understand, and seeing that it's a problem that needs to be solved and solving it. And that's what the next talk is about. It's a strange one. So Vincent Savalainen is our professor of organismic biology. He's not here today. He's famous for his work on sympatric speciation in palms. He's very much a genomics guy. But he's interested in something else. He's interested in the evolution of homosexuality. I say it's a strange topic. It's one of those things which is a phenomenon which is very widespread, evolutionarily inexplicable, and a happy hunting ground for crank theories and politics. It's not a subject you really want to tackle at all, but Vincent thinks that it needs to be tackled, and I think he's absolutely right. And if he does so successfully, it will be in good part, because Jackson Clive is working with him. Now Jackson, his biography is terribly simple. He's done his entire education here at Imperial, except for a short break out in the Max Planck. And he's going to be talking to us about his studies on macaques.
Hi, everyone. Yes, I'm going to be talking about my research into same-sex sociosexual behavior, which we conveniently abbreviate to SSB. Now, SSB, which can be broadly defined as non-reproductive sex between individuals of the same biological sex, has been reported right across the animal kingdom. So we know it's common across species, but we still don't really know how common it is within species. But there's an even bigger question here, which is, can the behavior be heritable? Is it capable of evolution? Now, theorists of yesteryear have said no, and sought to dismiss the behavior as a pathology, an aberrant consequence of extreme environmental conditions or even captive conditions. Still others have said, yes, it can be, but this leads to a Darwinian paradox in which surely the behavior is carrying a fitness cost associating with trade-offs with reproductive effort. Still others are saying, yes, it can be heritable and evolve, and what's more, it can be socially adaptive. It can actually carry fitness benefits. So all these and many other theories are all resting on this big question, can the behavior be heritable? So what do we know about heritability of SSB so far? Well, there's some evidence in two rather distant systems, in invertebrates and human beings. But each of these systems has its caveats. In invertebrates, research is suggesting that actually the behavior is an altogether different phenomenon underpinned by indiscriminate mating system strategies. And in humans, there's always this issue of self-reporting bias and the cultural stigma surrounding the topic. In this example, an absolutely brilliant genome-wide association study of over 500,000 individuals, heritability of SSB was found in both males and females, but there was also this cultural effect whereby older individuals were reporting a lot less lifetime same-sex activity. So to investigate this question, we turn to a colony of rhesus macaques living in Puerto Rico, which
quite remarkably, over 60 years ago, there was some sort of observational data that suggested that same-sex behavior in these males of this colony was actually almost as common as its counterpart, different sexual behavior, the males and the females. This observation was pretty much ignored in subsequent decades, but we thought we'd go back there and take a look and see what's going on. So we spent 3 years with this colony building behavioral profiles of same-sex behavior in males across 2 social groups. And the colony itself has been managed for over 100 years, and there's been lots of research into making sure that the population genetic structure and the social group structure is exactly equivalent to fully wild populations found in South Asia. And what we found is quite staggering. We found that males mount males, this fundamental mode in which same-sex behavior occurs in these animals. Males mounting males is at least, if not more common than males mounting females. The left-hand graph is essentially showing that more males engaged in same-sex behavior than engaged in different sex behavior, with the majority engaging in both. The right-hand graph is saying that it's showing that we found that for our sample males, our 236 sample males, their same-sex activity was almost twice that of different sex, although these numbers became more equivalent once we were only counting the mounter, the male that is on top, however you want to call that, and not including the mountee, the male on the bottom. On to the million-dollar question. After accounting for other effects, social group structure, sex ratios, individual age, social dominance rank, we account for all of that. The end point is, yes, the behavior is heritable. Now, we did this using the colony genetic pedigree records to infer relatedness and its subsequent effect on SSB between individuals. We also found a genetic correlation between the mounter and mountee behaviors. This is actually a fascinating new piece of the puzzle, I think, because developmental biologists have long been anticipating that we'd expect a negative correlation between these two things, the behaviors being mediated in part by natal androgen exposure that's masculinizing or feminizing different morphs and different subtypes of SSB. So this is quite an interesting result in its own right. But on to this question, so we know it's heritable now, but we go on to this question of fitness cost. Now, the first thing to say is that when we modeled our behavior and extended the probabilities, the longer we sampled our males, the more the likelihood was that they're behaviorally bisexual. So that's the first port of call. If they're behaviorally bisexual, we don't anticipate these massive reproductive costs because there's still some reproductive opportunity. But we can go one step further, or we tried to, and we used the genetic pedigree to actually look at offspring side per individual, and after adjusting for differences in age and the rest of it, we found a positive trend, non-significant, unfortunately, but at the very least, there was clearly not this sense of a strong reproductive cost to increasing levels of the behavioral activity. Perhaps with more data, we could get a more robust result here. But this, at least, this positive trend between same-sex behavioral activity and offspring side, it actually opens up the door to some of these socially adaptive hypotheses that we've heard about. You know, there's the examples with the bonobos and sex-mediating central tension regulation in the groups and stuff like that we've sort of heard about in the news. And we found, that conflict coalition partnerships were mediated by same-sex partnerships. So the individuals that did these behaviors together were also more likely to support each other in aggressive interactions with other individuals. This is the first time this has ever been shown in any system, and conveniently, a study this year showed in a closely related species, the crested macaque, that this coalitionary partnership trend is actually associated directly with fitness benefits. This has led us to consider terming this hypothesis the sacred band hypothesis, which is after the famous sacred band of Thebes of antiquity, a real battalion, Greek battalion of history, in which all the members of the platoon were male lovers and were said to fight extra ferociously and bravely for the sake of their partners. So to summarize, the males mounting males is at least as common as males mounting females. The behavior is heritable and therefore opens the door to all these evolutionary hypotheses, at least in macaques. There's no sign of reproductive cost, and especially when we consider that it appears that they're essentially all behaviorally bisexual. And finally, that there's this possibility of an adaptive function via strengthened conflict coalitions. So just some acknowledgements, especially Armand's already mentioned. My supervisor, Vincent Savalainen, had a lot of involvement in this project. Thanks very much for listening.
One of the things that characterizes nearly all of us who work here at Silwood is a love of nature in all its glorious diversity. It's just an aesthetic thing that is deeply ingrained in us. It's therefore important and unsurprising that conservation biology has always been very strong up here at Silwood. We all grasp the depth of the biodiversity crisis. The thing about conservation biology, conservation science, is that it often seems to sort of footle around at the fringes of the problem. The science is terribly interesting, but it often usually doesn't actually change anything. There are some exceptions, however, and the work of our reader in conservation ecology, I think I have the title right, Christina Banks-Leite, is an example of it. She has been working on the Atlantic rainforest of Brazil, and her work there has had a direct impact upon shaping legislation to conserve one of the world's most important terrestrial biotopes. The next talk that we're going to hear is by Jenna Lawson, and it's a very similar story, except it's not about the Atlantic forest of Brazil. It's rather in Costa Rica, and it's about monkeys, but it's another example of how beautiful work in biology, in conservation biology, can result in actually a direct impact. Jenna did her, let me just look at this here. Bachelor's degree at Keele, Masters at Edinburgh, and it was, of course, a PhD in Christina's lab. She's now, of all places, in aeronautical engineering in South Ken. It turns out that's very strange, but it's not that strange. She's figuring out how to use drones to put remote sensors at the top of jungle canopies. Jenna.
Hi everyone. So today I will be discussing my PhD research that I did with Christina. And the research was using passive acoustic monitoring and AI to conserve the endangered Jeffrey spider monkey in Costa Rica. So I think we can all agree that we're in the midst of an ecological crisis. Land is being increasingly altered by human activity, and more species are at risk of extinction than ever before. Now, monitoring and evaluating the response of species to anthropogenic change is essential for improved decision-making. However, despite decades of research, and as stated in an article by leading experts only in 2020, we currently lack the evidence to effectively protect and manage many vulnerable species. Now, this lack of evidence is due to many things, including failure to collect data at wide enough temporal and spatial scales, and also not evaluating the effectiveness of our management actions. Now, in this study, we used conservation technology, including passive acoustic monitoring and AI, to collect data at wide spatial and temporal scales to understand tipping points of human disturbance in the Jeffrey spider monkey. So the use of passive acoustic monitoring was central to this study because it allowed us to collect data at scales previously not possible with traditional sampling methods. And the development of an automated detector to actually analyze this vast amount of data allowed us to analyze 60,000 hours of data in just a few weeks. Now, if I was to listen to that manually, it would have taken 30 years, which was well outside of the realms of any PhD research. So I just wanted to play for you the call of the spider monkey, which is called the whinny, which was what we developed the detector for. It's very loud. And if we look at a pictorial representation of this, which is called a spectrogram, this is the image that we see here, where we have time along the x-axis and frequency along the y, and then the lighter the colours, the stronger the amplitude. So this is what a pictorial representation of the spider monkey's call would be. Okay. So just to introduce you to the study region and some of the conservation challenges that we were facing in this region. So my study took place in Costa Rica, a very special region in Costa Rica called the Osa Peninsula, which is on the South Pacific coast. And you can see on the left-hand side this map here. It's a land use map. And from looking at the amount of green, which is old growth and secondary forest, we can see that this is a highly forested area. And then the other colours, we have purple and blue, which represent mangrove regions and wetlands. And then we can see the red, orange and yellow, which are palm plantations, teak plantations, and grasslands. And then if we look at the map on the right-hand side here, this is a map of the protected areas in the region. So this area here in black is Corcovado National Park, a highly protected national park in the region. And then here we also have another protected national park, Piedras Blancas. Now this one up on the top is a protected wetland and mangrove region. And then in the center, this very light gray region is called the Golfo Doce Forest Reserve. So this reserve was put in place about 40 to 50 years ago, the idea being to connect these parks together, because it was recognized that due to a long history of land use change and hunting, that all the species in the area were located within this bottom black area, Corcovado National Park. So they put this reserve in place. However, almost 40 to 50 years on, for a lot of species, there's been little dispersion outside of the area. And this hasn't been studied by science, but this is what rangers and local people thought was happening. So it was our aim to come in and understand why this was taking place. So we decided to study the Jeffreys spider monkey. The spider monkey was considered to be one of the worst affected species in the region. And as an arboreal forest specialist, management actions that we put in place to protect this species have the potential to have a positive impact on many more. So we installed almost 400 audio recorders over a region of around 10,000 hectares. And all these white points that you can see on both of the maps were the audio recorders. The idea being that if we heard them on the audio recorders, they were present, and albeit some detection issues, if we didn't hear them, then they were likely absent. So what did we find? If we look at the map on the left-hand side, it's the same land use map with the points on, and in white we can see the points where the audio recorders, where the spider monkey was found. And if you look at where these points are, we can see that the last known location of the spider monkey is somewhere around here. And actually, the local people were right. They haven't really dispersed far outside of Corcovado National Park. But why was this? So the first problem was land use. If you look at the graph on the left-hand side, we're looking at occurrence probability in different land use types, and we can see that we only found the spider monkey in old growth and secondary rainforest. So they're highly dependent on forested habitats. We didn't find them in the palm or teak plantations, in mangroves, or in grasslands. And then even more interesting, within these forested areas, we actually didn't find the spider monkey until we reached 80% forest cover in a region, which is incredibly high and shows what a forest specialist this creature is. And then if we move to look on to the effects of roads, this map on the right-hand side is a map of all the roads in the region, which was hand-drawn in ArcGIS. And the yellow roads, the yellow lines represent gravel roads, so non-paved roads. And the red roads, which is the main one, is it's a bit difficult to see, running all the way around here and down here. This is a primary road, a larger paved road system. And then if we look at the graphs on the the left-hand side, the one at the bottom is looking at occurrence probability with varying levels of gravel roads. And we can see that the spider monkeys, they're not too bothered by gravel roads. There's not a large effect. However, what was very interesting is if we look at the graph on the top left, we can actually see that we didn't find the spider monkeys within 1 kilometre of paved roads. So they really, really are reacting strongly to paved roads. And then finally, what we found is that we obviously knew that the spider monkey was going to be in Corcovado National Park. We also found them in the wildlife refuges, which is sort of little areas like this, and these are considered even more highly protected than the national parks because they're protected by hotels who are protecting them for tourism purposes. We didn't find them in unprotected areas. We didn't find them in the wetland regions or, interestingly, in Piedras Blancas, and we didn't really find them in unprotected areas. So there's a high effect of how strongly an area is protected. So now we knew where the spider monkeys were and we knew where they weren't, and we knew exactly what was controlling this. But we wanted to provide a spatial component to our analysis to help decision makers in the area as a management tool, especially because it looked like from the results there was an issue with connectivity. So if we look at this map on the left-hand side, this is called a habitat suitability map, and this tells us where is suitable and where is unsuitable for the spider monkey to currently live. So if you can see on the map, all of the green areas is considered suitable. So this is linked to areas where it's high forest cover. So we can see Corcovado National Park, which is here, is highly suitable. Piedras Blancas is highly suitable. And there's a lot of suitable land in the middle. But if we think back to where we last found the spider monkey, which is around here, it's actually correlating to an area where we have road systems, but also a large area of grassland, palm plantation and teak plantations. So there's obviously an issue with connectivity at this particular point. However, even if the spider monkeys can get past here, what they've got to do is navigate all this section through here, which is more palm and teak plantations and grasslands. And then the big problem is this big paved road that's running around here. So we've learned from this that the reserve that was put in place to protect the spider monkey initially and all the other species in the region, it's currently not functioning as effectively as it needs to protect these species and to connect the two national parks together, as was its original aim. So in terms of the impact that this study's had, I'll turn that first quote on its head and say that I think from this study that we actually have developed the evidence to effectively protect and manage many vulnerable species in this region. Now, we can develop that evidence, but now it's up to the people in Costa Rica and in the local region to actually put that into practice. And I'm going to tell you a little story while I play some lovely sounds of the rainforest, maybe a bit quieter than before. So last year, unfortunately, the Costa Rican government decided they were going to pave a very big road straight through the middle of the region, and this would have been a development that would have had disastrous consequences for the spider monkey and for every other species in the region. If you remember the effect of paved roads that I told you about, with them not going within 1 kilometre. But using this evidence about the effects of paved road on spider monkeys, we were actually able to halt that development taking place alongside community groups and NGOs. Now, unfortunately, just this week, that development was put back onto the table, and I've been invited to go out to Costa Rica next year to actually discuss this development with the local government, and hopefully we'll get that development taken off the table for good. So, thank you.
I suppose the thing that really strikes me when I listen to this talk and when we were choosing them is how much ecology and evolutionary biology have transformed in the time that I've been here. That it's from a science which had a certain esoteric nature to a science which is now fundamentally about changing things in the world. It's become an applied science as much as anything done in South Kensington. And I think all these talks in one way or another illustrate that perfectly. We wanted to think about the future, what the next 75 years of science will bring for ecology and evolutionary biology, and for Silwood specifically. Now, of course, the future is inherently unknowable, but I was inspired to think, in doing this exercise, by an essay written by J.B.S. Haldane, the famous population geneticist, in 1923, called Daedalus, which is explicitly an exercise about speculating about the future, and there's amazing things in it. I mean, Haldane was a brilliant man. He discusses the possibility that in the future, England will be covered with giant metal windmills, which will be hooked to electric motors that in turn will generate electricity for the national grid. He says that's the most obvious place where we can get our power from. And he says, I think it is perfectly feasible that we will see this technology in place in the next 400 years. Right? So, prediction, it's a tricky thing. Who better to predict the future than Yadvinder Malhi? He is a professor of ecosystem science at the University of Oxford and director of the Leverhulme Centre for Natural Recovery. But most importantly for us, he is the president of the British Ecological Society. And indeed, it is this very week that the British Ecological Society has been having a forward look about the future of British ecological science. And he's here to give us the results of precisely what Britain's best and brightest ecologists have been thinking about exactly this subject. Now, to help him predict the future, we've asked a few of our own staff to join him in a panel discussion. So first is going to be Yadvinder, and then we're going to get his colleagues, some of our colleagues up here. There's going to be Guy Woodward, who you've met, Tom Bell, Professor of Microbial Ecology, Christina Banks-Lighty, Reader in Conservation Ecology, I've already mentioned, and Will Pearce, Senior Lecturer in Applied Ecology, and then they're going to spend about half an hour telling us about what they think the future will bring. Yadvinder.
Do you have a clicker? Okay. Thank you. Presume this works. It's a pleasure to be here. Thank you for inviting me, and I've really enjoyed the session. I've been fascinated by Mick's history of this place. I've learned a lot about things that I vaguely had perceptions of. And the young researcher talks are absolutely all uniformly brilliant and really insightful. So thank you for your contributions. So in my role as president of the British Ecological Society, we seek to be a convening organisation for the ecological community in the UK. And among our roles is to perhaps help navigate or chart the future pathways, which is why we had this workshop looking at the future of ecological research in the UK. And I'm going to give you not the answers that Armand advertised there, but I think perhaps some of the processes that we went through when we had a group of people together, and some overall perhaps structure, some of the things that we're thinking about. But this is a process that's ongoing. It isn't a fait accompli with that particular workshop. So it'd be great to have your thoughts afterwards. Feel free to email me or talk to me in the reception about that. And so this was a workshop that we had just on Monday and Tuesday this week in London. There were about 40 of us there, about 20 academic ecologists, and also 20 people from the policy or the stakeholder world, whether from Natural England, whether from the ecologists in the national government or in the devolved governments as well. So it was a mixture of academic ecologists and ecology practitioners of various forms. And that also shaped those debates in quite a strong way. Here's a picture of at least a majority of the participants from a range of these sectors. And in some ways, it's a bit of a fool's game. Get 40 ecologically trained people into a room and get them to try and agree on anything. And as you know, you're not going to get very far overall. But I think we had some interesting conversations and some overall structure of where we were seeking to go. And this is one of the outputs, which is my colleague Cécile Girardin and Adisi Key produced this as an ongoing project doing that. And partially it captures some of the key debates we were having there, some of the things that emerged, but also captures the potential of chaos in this system as well, in terms of trying to chart the future. And this is this is the second start of a process. We had a community consultation back in January, February, where we asked anybody, whether in the BES or outside, to just give their thoughts about what they thought the priorities were. And we had a whole range of suggestions. And then this part of the workshop was trying to convene an overall architecture out of those range of suggestions. So I'll delve a little bit into some of the things, some of the words that you're seeing in this chart later on. But before I do that, I want to delve in some of the key debates or tensions that we came across when we were trying to think through this. And I'm sure that we got this diagram. If we randomly selected 40 different ecologists, I wonder how similar or different the diagram would be. So clearly it's a product of the people in the room and the ideas that are there as well. But one question that came up, what is ecology for? Oops, let's go back one. Or another way of framing this is ecology a normative science? And just to explain that, a normative science is one that's driven primarily by its values, its value system. So medicine, for example, I think could be described as a normative science. Its primary function is to make people better, and it does lots of amazing primary research and discovers how the human body works, etc. in the process, but its ultimate goals are value-laden. Astronomy, perhaps, is the archetypal non-normative science, where it's driven ultimately by a fascination in the way the universe works. There can be interesting spin-offs and technologies that benefit humankind, but its fundamental goals are not value-driven. And where is ecology? And in some ways, some of you may be groaning in time, because obviously the answer is very obvious. It's both. It's driven partially by fascination with the nature of living systems and interaction of living systems and how those systems both evolve, assemble, function, change in the world. And that's the fundamental questions that we're interested in. But also, it is driven by normative values around trying to address issues, whether it's addressing the biodiversity crisis or malaria or COVID-19 or other issues. So there's an element of both. So this may seem like a silly question to ask, but when, as a community, we're trying to come up with suggestions or guidelines, it's important to be aware of this, because in a room full of practitioners, it could be that all our suggestions are to do with a normative question like addressing the biodiversity crisis. But is there a danger if we come up with recommendations like that we forget the fundamental intellectual priorities of ecology that help inform those more normative questions? Similarly, in a different audience, maybe full of very academic ecologists, we may focus on those intellectual questions, and those normative products will be seen as products of those intellectual questions. So, and if we ended up, if our recommendations ended up on the desk of NERC decision makers, it may have consequences whether we think of it as a normative or a non-normative science. So that's one thing that we had to think through. And in that particular room, with a lot of policy implementers in the room, a lot of the decision making was driven towards normative questions. The second one is what is the space? If we agree that there are normative values and questions in ecology, what's the space for ecological theory to fit into that? And there are many different diagrams you could make. Here's one inspired particularly by some contributions from Jabouri Ghazul. You may or may not agree with this, or may see your particular area in here, or may see that it's not in there, there. But this is one example of how we could have an integrated theory of change in ecological systems. I won't go through every part in detail, but a mixture that looks at population dynamics, network dynamics, how ecosystem interactions with these lead to functions and services in ecosystems, and then how those ecosystems reconfigure, the evolutionary dynamics of that, how that leads to system-level dynamics such as resilience and vulnerability, the role of spatial and temporal heterogeneity there, and the link with external drivers and biogeochemical dynamics, and then to what extent the drivers of change, global change, social, ecological, or economic systems affect this, and how those are affected by cultural change or by norms, and then various broader contexts of ecology in terms of the changes that we're seeing in ecosystems in terms of biodiversity, agriculture, other uses of the natural world. So that's one possible framework for thinking about the whole range of ecological theory, and there are many, many other ways of doing it. So that gives a place for the theory of ecology that can be applied to more the normative questions, but also is driven by that theoretical interest. But then if we agree that influencing issues, influencing, say, in particular the biodiversity crisis, and we want ecology to do that, how can ecology be most influential for policy? And to do that, we need to have some understanding of a theory of change or a theory of mind for ecology and how it works to influence policy. And here's one example of this. This is work largely derived from work by Claude Garcia at Zurich. And when you're trying to influence a scheme of policymakers, there's a range of questions you can ask. And one is, has the person you're trying to influence heard about the issue that you're concerned about? And if they may not have heard about it, in which your job as a scientist or as a policy influencing scientist is to inform. And that's probably where the majority of academic research goes, in trying to inform the public or the policymaker about issues such as climate change or the biodiversity crisis. But if they have heard of that issue, the second one is, do they believe in that issue? If they're not, they can be classed as a denier. They simply don't believe the evidence that you present in front of them. But if they do, then the third question, which is a bit more tricky, is that yes, we accept there's a climate change, or we accept there's a biodiversity crisis, but how do we trade this off against all the other things that I have as a decision maker or a policy maker in terms of food security, for example, or economic growth, and things that are particularly salient in discussions this week. And so either they're to occupy to make bandwidth, even if they believe that this is an issue, it doesn't reach bandwidth in their busy decision-making minds, or if it does do that, it doesn't play off against the trade-offs that they're making, whether in terms of broader issues as food security or immediate issues like electability. And so that's another area that we have to do. And if they agree it's a priority, then it comes to agency. How can we make a difference, whoever that your target of your work is? And it can be that many issues, many members of the public can feel concerned but powerless, unable to tackle the gravity of the situation and feeling and making a meaningful difference. Or they can feel that they can contribute in some way to that, and in which case they can be architects of change. Now, I would venture much of ecological science interaction with policy is in this area, just in simply in the information side. But another key area is often around the architecture. And so we heard some examples today in the talk. So, for example, with Costa Rica and the decision about the expansion of the road, that's an area where the research is influencing the architecture of policy-making decisions. But all of these require different types of translation of ecological research into something that works. So information, belief systems, values, and strategies for change. We tend to focus on those two, perhaps, but a lot of the other work we do in simply describing the wonders of the natural world, the complexity of the natural world, are fundamentally important in shaping the essential ones. So what are the predominant belief systems and values of the public that can help be agents of change in the longer term? So, one of the challenges is whether we short-circuit this and sometimes also get things from the information stage to the architecture stage at an easy area, in an immediate form. So then moving on to that melange of discussions that we had and our attempt to come up with an overall narrative. What we didn't want out of that process was simply a top 10 list. That as a community, we did a ranking and we voted, and this is our top 10 league of priorities. What we wanted to do out of those ideas is to necessarily pick up some things that we felt were organizing priorities that were emerging in the discussions, but to have a narrative and an architecture around them, a story to tell. The storytelling is also important in trying to influence change and trying to shape agendas. And so these are our 5 attempts at overarching themes. This is very much a work in process. We're going to send this out for consultation and then get some thoughts and feedback in that, and also in examples. So this is based on some of the ideas that are in here. And so to go through them, oops, let's go back one. In each of these, let's go back. Great. So the overall theme is at the top left, and that's where we've agreed on overarching architecture. The things in blue boxes are just examples in there, and ultimately we may decide on selecting 3 or 4 examples as priorities, but this is something that's still very much up for suggestion, contribution overall. But one aspect that we found was a particular organising theme was around novel futures, thinking about the new futures for ecological systems in the context of global change, of invasive species, a changed climate, and recognition that ecology has moved out of the ecological systems of the Holocene into whether you call it the Anthropocene or something else, and the new nature of ecological systems in a rapidly changing planetary system. And themes that emerged in there are things such as novel ecosystems, the new native species and how they interact, nature recovery, rewilding, novel climate regimes, and also how to build climate change adapted ecosystems. And this idea of shaping novel futures for ecology that are less based on historical compositional baselines, but look at ecological processes and how they can be made resilient and vibrant in a transformed future was one area that was recognized. A second theme, very different in concept, was what we call learning by doing. And this is a need to just get out there and do things on the ground and learn by experimentation, learn by the mistakes that are made in experiments as well, rather than having too much debate about what should be done, just going out and experimenting with systems in terms of restoration, in terms of recovery. Particularly, a thing that was picked up was the idea of living laboratories. And in some ways, I think some of the work that Mick was describing here of the last 75 years is exactly an example of living laboratories of long-term change, exactly the sort of things we need to see happening in the wider landscapes. I should also mention, I didn't mention at the start, the focus of this workshop was priorities for ecological research in the UK. And I think many of the themes that emerged are also relevant for global ecology. And our hope is to do something similar in terms of global ecology and global ecological questions, because actually a lot of the things that emerged are generally applicable to ecology, not just to the UK-focused ecology. Next generation biodiversity indicators, some of the innovation that's coming in technology and other approaches to look at indicators of biodiversity, issues around data standardization and benchmarking biological health, ecosystem health were some of those issues that were discussed there. The third one was around the dynamics of ecological change of ecosystems in terms of ecological collapse and resilience, extinction and speciation dynamics, the multiple timescales of dynamics in ecosystems, how emergent behavior appears when considering these. Particular themes such as the cumulative impact of repeated stresses, and also technological aspect there in simulating systems to understand these dynamics and changes over time. And a fourth theme was a word called wholescapes, which was first suggested by somebody from WWF, and we all groaned and thought it was a terrible word. And then in the end, we couldn't come up with a better word, so we stuck with it overall. And this is the idea of going beyond the ecological boundaries of your particular system of focus and thinking about the wider scale interactions, because in solving things such as the biodiversity crisis, it's all about the system level the changes in which the ecological system sits. So an obvious one is the triple challenge of reconciling ecological values with food security and climate change and other factors, understanding the nexus of that in real landscapes. The multiple values of ecological systems, whether it's carbon or water, or whether it's cultural values and social values, and finding out ways of valuing nature in our economic system, in our global decision-making system. Global telecoupling and offshoring, how things cascade economically or physically. So if we decided to rewild half the UK, what does that do to our food system? Do we end up importing our food from Brazil and causing negative biodiversity consequences elsewhere? So understanding those connections. Urban-rural connectivity, the role of urban landscapes, and particularly that as a primary interface for most people with ecology, and how that fits into the broader landscape. And also a whole scape in the sense of not focusing on terrestrial ecology or aquatic ecology or marine ecology, and looking at the connections between those systems to have a more integrated geographical ecology. And the final theme was around discovery, that there are still lots of things in biodiversity and ecology that we simply don't understand. And there's still many frontiers of discovery in this realm that we should prioritize. So that ranges in hidden biodiversity, such as the biodiversity and functioning of soils, of forest canopies, the deep biosphere in sediments. There are many other examples. The ecology of pathogens and novel pathogens, in both how they emerge and then how they can be understood and managed. The impacts of novel pollutants, novel chemical factors, and how they affect ecosystems. And I think a strongly emerging field is the ecology of human health and well-being, in both ways, both in terms of negative impacts, such as we saw in the pandemic, but also a variety of positive impacts ranging from mental health and well-being, but also the microbiome, the volatiles in the air. There seems to be the visual spectrum of how people respond to ecosystems, and that if we can align parts of the ecological agenda with the healthcare agenda, I think there's a huge potential there for growth. So just to sum up, as I said, this is a work in progress. This is certainly a workshop that didn't try to define these questions. It almost tried to define the types of questions that we're asking. We have a draft report that we'll get out in the next month or so, and very open to consultation and feedback and thoughts. And a final report early next year for dissemination across communities and stakeholders, and discussions with stakeholders after that, including funders and other options, to help chart at least the next decade or two in terms of what we think as a community the priorities should be. So thank you. So I think let's invite the panelists onto these chairs, and then I think we'll just work through
because otherwise they're not going to see us.
Can you see on the camera? Do you want it to go up? Yeah.
Up. Let's hope nobody falls down.
Exercise. I'm trying not to do. Just careful where you put it.
Okay. So I won't do introductions, but I'll just, I think, hand over to starting with Chris.
Thank you. Oh, can you hear me? Yeah. Thanks, Yadvinder. Yeah, so when I was thinking about the next 75 years, trying to really think how we can apply the ecological knowledge to policy, to change the environment, and to help people, really, for the benefit of society. And there's mainly two ways that we can do this. So one of them is to work closely with policymakers. But as Yadvinder was saying just before, there are several layers of potential issues, and one of the layers sometimes is, do I believe in these changes? So despite the fact that in the past I've had a reasonable success managing to transforming some of my results into policy in Brazil, so we managed to base the results were used to base three different pieces of legislation in Brazil that then helped the protection and restoration of the Atlantic forest. And I know that many other people here have had plenty of success as well. I've got to say that I don't know to which extent today I would have had the same success, because my results would have stopped probably at the, do I believe in this? Do I believe this is necessary? But so while I do think this is a reasonable issue, I also started seeing the future with a bit more hope, because the other way that we can also influence the environment and people is to work with businesses. And there are some companies now, the big polluters, the ones that produce a lot of carbon, they are waking up to the crisis we're facing, and some of them are moving to net zero or sometimes even to carbon negative policies. And they are pairing up with companies that are rolling out large-scale, high-quality reforestation that can have really good co-benefits for biodiversity and to also help local people. So myself and a few others here in Silwood are working with these sorts of companies now. And personally, I think that the future, at least in the near term, is going to be very much marked by ecological science influencing policies within businesses. So that's it for me.
Thank you, Chris. So I thought my area of research required a little bit more introduction. So I work on some of the least noticed, some of the least charismatic organisms. These are the bacteria. So if you go into any habitat, look on any surface, then you'll find bacteria there in unquantifiable numbers. Every time you take a nibble of cheese, sip of water, you'll be consuming hundreds of bacterial cells. Every time you shake hands with someone, then that will be a dispersal event. They're incredibly abundant, they're incredibly biodiverse. To a first approximation, all of the biodiversity on Earth is bacterial. And not only that, they're incredibly important. So if you think of all the biogeochemical cycles that we're interested in, flows of carbon, of phosphorus, of nitrogen around the world, all of them would be halted if the bacteria disappeared. So it's, I think, an incredibly important group of organisms, and it's a group of organisms that we've been studying for some time now. But I think it's fair to say that for the past 75 years, we've been somewhat groping blindly amidst the mazes of the ultramicroscopic. But that's all going to change, and in fact that's in the process of changing now. Because of advances in omics technologies, we've been able to really accelerate that kind of research. We have genomics, we have transcriptomics, we have proteomics, we have phenomics, we have all the omics, all the time, everywhere. And all of that has allowed us to, for the first time, in the last 10 years really, to characterize these communities for the first time. We can now go out into a soil and we can say what's there, we can get an idea of what they're doing and how they're influencing all of these really important functions. And that's something that's happened very recently. At the moment, I think we're in sort of this Victorian era of discovery, a little bit like Darwin and Willis, sort of going out and seeing what's there. For the next 75 years, we think, well, given all the changes that have happened just over the last 10 years, it's probably a fool's errand to try to predict this, but Guy emailed me a couple of days ago and said, okay, come up with a plan for the next 75 years. So I think what's going to happen in the next 75 years is that there'll be this continued acceleration in the pace of discovery within these sciences. Unlike for these poor people who are studying things like spider monkeys, tropical trees, and so forth, we can actually observe population dynamics in a few hours. We can look at community dynamics in a few days. We can look at evolutionary processes in a few weeks. And so all of that means that as long as there's concerted effort, we can really start to understand these systems over much less limited times than is the case for my ecological colleagues studying larger charismatic megafauna. And so I think what will happen over the near future, over the next 10, 20, 30 years, is that we'll gain a really in-depth understanding of how these systems work and be at the forefront of ecological research. We'll go from sort of catching up with understanding the ecology of these systems to really leading ecological thought over those sorts of timescales. 75 years is a long time in the future, and I think for those sorts of time horizons, we'll have reached a level of understanding where once you have the understanding, then you can start to do the manipulation. So what I think we'll be able to do in 75 years is to be able to tinker with these systems so that if we want less CO2 in the atmosphere, then we can modulate that at a global scale. At a local scale, I think, if we want to grow a plant better, then we'll be able to manipulate those soil communities in order to grow plants better. And I think probably, our ancestors or our looking into the future, they'll look back at our ancestors, at us sitting in this room in this sort of soup of undescribed microbes. They'll probably look back in horror and say, how could we do such things? How could farmers just throw a plant into the soil and think, well, just expect it to grow without actually manipulating what's there? But I think the answers are literally on our fingertips.
Cool. Thank you, Tim. So when I think about the next 75 years, I think a little bit about what the last 75 have looked like. And I think in ecology, we've done an amazing job of responding to some of the great challenges that have faced us, okay? We've seen a war coming, and so ecologists have thought, okay, how can we store food? We've seen pandemics coming, we've thought, how can we control them? We've seen populations growing, we've thought, how can we grow more food? And I think we're at a really amazing moment right now where ecologists, finally, what we care about is understood for the importance it is. We have lots of terms for these things, don't we? Ecosystem services, natural capital, nature-based solutions. I like to think of them as things we can eat, things we can drink, and the air that we breathe. And finally, those things are being recognised for being important. So that's wonderful. But the reason it's being recognised for important is that we're at a crux moment. And if we don't do something now, and I would actually argue that the next 75 years of ecology will be determined by the next 25. Because if we do not solve the problem of how to have air, food, and water, while also having biodiversity, if we do not solve that in the next 25 years, I don't actually know what the 50 years after that will look like. So I think if we are going to come up with the tools and the ability to deal with those problems, we need to start making ecological forecasts. We need to start giving policymakers the tools that they need to actually solve these kinds of problems. And so to do that, I think we need three kinds of things. We need a new kind of data, we need more generalizable models, and I did have a different word for this, but I'm just going to steal what Armand said. We need to be intellectually promiscuous, okay? And so I'm going to talk about what that might mean. So in terms of data, when I think about the last 75 years, we have nailed the ecological experiment, okay? When I think about Silwood Park in particular, we've had some absolutely fantastic global scale experiments that have been applied here, and there is still a very important role for those data. But when I look to the next 75 years, I think we're going to have to start looking towards huge scale observational manipulations, kinds of things like the SAFE project, perhaps like the Ecological Fractal Network, that are distributed across the globe and are connecting all kinds of data, observing the entire system. Because ultimately, if we want to solve some of these grand challenges, we have to understand that system, which means we can't tinker with it, because the system is us. We cannot run an experiment on us without, being really quite evil. So we're going to have to start really changing the way we collect global data. We also need to start thinking about how we can generalize our models. It is not a surprise to people that the evolution of species matters. And I think the next generation of models over the next 25 years are going to think not just in terms of counting species or numbers of species, but the kinds of species, how those species have evolved, the conditions that led to them that will allow us to make models that can generalize across regions, across time spans, so that we can finally start once again making forecasts wherever and whenever we need to in the world. And then finally, promiscuity. We're going to need to be intellectually promiscuous, okay? When I think about the talks that we just saw from some of the emerging leaders that we just saw earlier, I see people who are really delving very seriously and very carefully into other fields. I see microbial ecologists who are saying, not just how can I use Google mobility data to understand where people are moving around, but are asking questions like, what are the drivers of how people are interacting with one another? What's the sociology underlying the system? I see people saying, not just how can I use bioacoustic monitoring to know what is somewhere, I see people really driving into the technology behind that. And so I see in the next 75 years, more and more ecologists being intellectually promiscuous, people being subject experts in two domains simultaneously. And so, really to summarize there, I think what we're going to see in the next 75 years is going to depend on the next 25. And if we can get that kind of data, if we can make generalizable models, and if we can start talking and learning from other disciplines, I think we really have everything we need to solve the grand challenges we have right now of feeding people, while also having a planet that is left for others.
Thanks, Will. Okay, so I'll just add a few little words here. So this morning, thinking about this in a bit more detail, I jotted down on this small bit of paper what I thought the next 75 years might look like. And really, in many ways, I'm just going to echo a lot of what has already been said. So the meeting that you described, Yadvinder, slightly different words, but many of the things that I've been thinking about are aligning with that. So it's kind of reassuring to me that I'm perhaps on the right track. But just to highlight some of the general trajectories, and I think exactly as Will says, you can learn a lot by looking back through the past 75 years and see the direction of travel. I think it's Niels Bohr, but I'm going to roughly paraphrase here. It said prediction is very difficult, especially when it's about the future. So I think that that's very true, but I think ecology needs to become a much more predictive science. So Will's touched on this and others have as well. We need to move beyond the generation of knowledge through to the understanding and particularly grappling with the mechanisms to then to be able to predict what future scenarios will look like. And if we can do that, then we can start to intervene, exactly as we've heard from others. And that's going to be really key. Now, to be able to do that, you need data and lots of it. And we're in the age now of big data. If you go back 20 years ago, we had a dearth of data. Now, in a way, we have a surfeit. I think maybe the bottleneck is the usefulness of some of this data, how it's targeted to particular questions. And I think we need to think about future experimentation and to target it much more carefully than simply kind of harvesting data and hoping something will pop out from the noise. So I think we need to think more carefully and be more nimble about that. I think we also need much better data and model integration. That's happening already, actually, and that will be a trend that's going to accelerate through time. We are working hand in glove now, really, much more so empiricists and experimental ecologists with modelers and theoreticians. So I think that that trend will accelerate. But exactly as others have said, it's not enough just to do it within ecology. So to meet these grand challenges that we face around things like food security, linking human health to environmental health, we started to see these things emerging, and the young generation here are showing the way with some of their research. Ecologists are going to need to speak with and work with much more closely medics, mathematicians, physicists, engineers. All of these disciplines need to be brought together. And I think just to sort of bring it back to the 75 years and the anniversary of Silwood, I think we're uniquely privileged to be in a position where we can do that. We have a lot of that expertise in-house, but we have also fantastic neighbours in our other campuses and the disciplines that we can work with, as well as, of course, the wider sphere. And that multidisciplinarity, I think, is going to drive the future. If we're going to become the weather forecasters of the natural world, which is where we need to get to be, and then the bioengineers of that world to fix some of these problems, I think that I think that's the direction of travel over the next 75 years. But luckily, I won't be around in 75 years to see whether my predictions are wrong. So thank you. That's my summary.
Okay, great. Thank you. Let's round of applause. Okay, I think we have a couple of minutes, so we won't have much discussion. But I was pleased to see that many of the points you made both fitted into the architecture and challenge bits of it, which is satisfying. So it's a point to think about. But Will, you made a very convincing case there that ecology is essentially a crisis discipline, that we've got 25 years to solve some major issues and challenges. So this is a question to anybody in the panel. If the BES is making a list of priorities, should the priorities be problems ecology needs to solve or things that ecology needs to discover? Does anybody have a pitch there that they would like to say?
You go first, Will.
Yes, sure. Yeah. Okay. So no pressure. Right. Okay. So I think that's a great question. I think the brutal reality is that. So one of the wonderful things about universities and about academia is that they value the idea that ideas matter, and that by learning things, we might have unintended consequences that might help, but we also might just find out cool things about the world. And so I think in much the same way that there's always going to be a role for an English literature department in universities, and there's always going to be a role for, pure mathematics and pure biology, I think, there is fundamental biodiversity research that matters and that we must continue. Not just because I think it will give insight into solutions, but also because it's cool, right? I mean, I think many of the people in this room, put your hand up if you like an animal or a plant or a bacterium in some, right? We all like them. Some people are about to start talking about fungi or something. Okay, fine. But, the important thing is it's beautiful, and so, of course, you must study it. But fundamentally, I think we need to start thinking about solutions. So I do think that matters, but we need to recognise that many times we already know the fundamental biodiversity theory to implement those solutions. We have to start connecting it seriously and carefully with other disciplines. It's not enough to just say, I'm going to be interdisciplinary because I'm going to talk to an economist. You have to really delve into the economic theory. You have to start thinking about why what are to an ecologist some straightforward solutions. Don't cut as much of it down. Leave it alone. You have to start understanding why those solutions are actually not as simple as they sound. And so I think the next fundamental research actually is not going to be pure research or solutions. It's going to be what are the intersections with other disciplines where we can find out why we can't do the things that we already think we should be able to do, if that makes some sense.
Thank you. Yeah. Anybody else want to pitch in some thoughts?
Well, yeah, I agree with Will in a way that I think often we already know a lot of the answers that we need. There are cases where specific answers are needed. So, for instance, Jenna showed an example where, to manage a particular area, we need a specific type of information, and that could help, preserve the biodiversity in that place. But yeah, so I agree also with Will that to reach many of these answers, we're going to have to step a little bit probably outside ecology and start interacting a lot more with scientists from other areas to reach a better answer, which was the case, for instance, with the work that Jenna was doing, because we wouldn't have reached that answer without interaction with people from the computing department. So it's potentially the solutions are going to become ever more complicated. I think the basic, we need to preserve things we know, but it's going to be tailoring the solutions to specific problems that it's going to be the challenge, and that is going to be achieved by interactions with others.
Yeah, I'd agree. It's almost a sort of slightly false dichotomy in a way, because the answer is you need both, and depending on the challenge, you need more of one than the other. But I certainly would reiterate the point that the others make. I think the biggest challenge we face as ecologists is to be able to speak to the other disciplines and to be able to engage effectively, because you spend an awful lot of the time when you engage on that multidisciplinarity, and it's a steep learning curve for myself, every time we do it, that you find that the languages and the approaches are very different. The way of thinking and actually even using the same terms means something very different. So even things like resilience, which we use in a very defined way as ecologists, would mean something rather different if you're speaking to, say, a town planner or an engineer. And I think that really trying to activate that nexus where you have these different disciplines around a challenge is where, oh, sorry, that's my phone, excuse me. That's going to be the real challenge, I think, trying to make the progress in those places. And you're going to need both the pure and the applied angles there. I don't think we have the luxury of prioritising pure fundamental ecology on its own at the moment. Equally, we don't want to become too prescriptive where we're really just almost kind of consultants dealing on a local case-by-case study. But when you have both together, that's when it's the strongest. That'll be my overview.
I'll be brief because I can see Emily at the back saying that perhaps it's time for drinks. But I think coming up with problems that need to be addressed, I think is a good way of focusing ecologists' minds. And I think if we can identify some of these grand challenges and get lots of people working on them, then that improves our overall chances of success. And often the blue sky stuff can sort of feed off of that, I think. Also just say, at least in my own field, that there's a little bit of a paradox between you have all this push for multidisciplinary research, but at the same time, individual researchers need to become increasingly specialized in order to understand their systems. And I think that's a paradox that we'll have to address.
I'll just add a final point to that. I think the key thing there is not to try and become a sort of Renaissance artist in the sense that you are the new Leonardo, where you are able to span all these disciplines. The depth of your own disciplines can be key, but the answer to that is to collaborate and bring in the experts who can do the bits that you can't. You just need to become more familiar with these other disciplines that you can speak their language, but increasingly it's going to be a team effort. The idea of the sort of lone genius solving the world's problems in their laboratory on their own is long gone. I think that the scale of the challenges and that need for multidisciplinarity means we have to work as teams now.
Great. I think it's a perfect point to end on. So thank you.
Thank you.
Panel.
We have a new head of department, Dan Davis. Except that we've known him for a very long time because he was at South Kensington and then had this meteoric career when he became professor of immunology at some absurdly young age, and then went to Manchester, and now he's just come back and joined us. I'm not going to say anything more about him, except, Dan, welcome back, and it's good to have you here. You've having just arrived.
Oh, thanks, Armand. Just get the photo off. Right. Okay, thanks so much. I'm hugely privileged and honoured to be here. I've been the head of the department for 7 days. It hasn't collapsed. This department still exists, which, as Andy Purvis mentioned in the coffee break, that can't necessarily be said of the leader of our country's record. I did my degree and PhD in physics because I thought that studying laws that work across the whole universe has to be the most important fundamental thing to study, and that's why I should devote all my energy to. After my PhD, I changed my mind. I really just thought that actually studying life is actually more profound, more important, and closer to tackling the really big issues of our time. I studied the immune system. Having seen today's talks, if I was a bit younger, I would perhaps change my mind yet again and move from studying the immune system to studying ecology, evolution, because it feels really, truly, deeply that the really big things that we face in society today, globally, are being tackled in the kinds of research that was presented today. And I'm not just saying that gratuitously. I mean, it's blatantly clear that conservation, evolution, ecology are profoundly important to everything that the world is facing. So it was a fantastic day. The other, there's another thing that comes to mind, which is that when Mick Crawley was presenting the sort of history of Silwood, which was absolutely brilliant, there's a lot of heroes of the place. And of course, we must rightly celebrate the heroes. There are also many heroes today. Armand mentioned many of the leading scientists that are here right now today doing truly, world-leading research. These are our heroes. But from my own world, we also have many heroes. And so, for example, as you know, the COVID vaccine was obviously profoundly important, and we have Sarah Gilbert and her team as an example of heroes that made that happen. And Sarah Gilbert planned a COVID vaccine in January 2020, and by November 2020, it was formally proven that vaccine worked. Unquestionably, she, as an example, and all her team need to be recognised as true heroes of our time. I think, for me, in a very real way, the happiness and solace you get from doing science is knowing that you are part of the community. There are heroes that we have to celebrate, and the example I gave was Sarah Gilbert's team, but for most of us, we're part of the community that also played a role in getting to those phenomenal big discoveries. What you have in Silwood feels to me like that you've, through the history of what you presented, it's very much all about community. And that is really a crucial part of what science is, that it's really about us being in the same community and we're striving to solve these big world problems. There are heroes that do the really final step that's critically important or have the big idea. but also we're all part of that same community, and it wouldn't happen without all of us working together as a community. So I think one of the great things that's happening here is that sense of community. One final thing, listening to this panel debate the next 75 years, brilliant, Christina, Tom, Guy, Will, Pierce, it was fantastic to hear that. It really feels to me, coming from physics, of course, I think it was 1887, Heinrich Hertz looked at, building on Maxwell's theoretical analysis. Heinrich Hertz finally saw waves that we couldn't see. There are lots of waves that we can't see. Heinrich Hertz, and he died at age 36. There's no way that he could have predicted that the waves we couldn't see would lead to the television and the internet and the radio and everything. So that felt like what you guys were talking about in that panel, that, we're working out the bacteria that are happening in soil, and we can't, in 75 years, it just feels like there's going to be profound changes of how that plays out in the world in which we live in, and it's actually impossible to predict how profound that's really going to be. So it was a huge honour to be here and see all of this, and if there's, yes, I'm the head of the department, but clearly Guy Woodward is your champion right here, and I'll do everything I can to help Silwood prosper over the next coming years, decades, and even longer than that. Please go and look at the exhibition, enjoy all the drinks, and also thanks so much to Armand, Emily Govan, all the team that put this on. Absolutely, what a phenomenal event. Thanks so much.
Ladies and gentlemen, thank you very much. Thank you, Dan. Well, it remains for me to personally thank my colleagues who helped me put this thing on. And again, the person who really needs to be mentioned is Emily Govan, our head of communication, and Katie Willis, who you've heard, she gave the mosquito talk. These are the two people who are probably most critical in doing it. I want to thank our speakers for giving us just a great afternoon, whether they were new researchers or the most distinguished. And finally, I want to thank you for coming out here tonight and this afternoon and helping us celebrate this amazing place and its past and its future. Thank you very much.